To say few lines about Sir. Sir is Professor and Head Department of Community Medicine, also Deputy Medical Superintendent, USIC Medical College, Rajajinagar, Bangalore. Sir has 15 years of teaching experience, and to his credit, he has got 15 national and 16 international, a total of 31 publications. Sir is a member of State Level Coordination Committee under National Iodine Deficiency Disorder Control Program, is a member of various other professional bodies and reviewer boards. Sir has served as Assistant Project Officer, Improving Access to Safe Abortion Care Services, Pathfinder International, New Delhi. Sir, being alumnus of Sri Devaraj Aras Medical College, has worked as proctor, convener, co-convener, member of various committees like Institutional Ethical Committee, Grievance Redressal Committee, College Development Committee, Student Welfare Committee, so on. To his credit, Sir has got various other projects ongoing, and Sir is involved in public health research, organizing conferences, workshops, has participated in various medical educational CMEs, workshops, so on. I welcome you, Sir. Over to Dr. Anil, Sir.
Yeah. Am I audible?
Yes, sir.
Yes, Dr. Anil.
Sir, first of all, I would like to thank Sri Devaraj Aras organizing team for giving me an opportunity to present on introduction to cancer and its global impact. My slides are visible?
Yeah, presentation.
Yes, Dr. Anil, you are audible. Slides are not visible.
Just a minute, sir. I can share it.
Share ಮಾಡಬೇಕು ಸರ್ ಮತ್ತೆ ಡಿಸ್ಕನೆಕ್ಟ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ ಈಗ ಮತ್ತೆ ಬಂತು
What about now, sir? Is it visible?
No, sir.
Not visible. No?
Your slides are not visible, sir. Network. Listen to slides, sir.
I think I'll off my video, then the bandwidth will be better.
Yes, sir. Okay, sir. Now it is coming.
Yeah, now it is visible?
Yes, yes.
Okay. So, is it coming? Can I move?
Yes, sir.
Okay. So I'll be going ahead with these contents, starting with introduction to cancer, classification of cancer, impact of cancer at global level, national level, state level, district level, local level, and the risk factors for cancer. As we all know that the normal cell division happens and new cells are formed as per the need. But what happens in cancer is the cells with damaged DNA also divide, but the proliferation which takes place is very uncontrolled, and the division also what takes place will be having a uncontrolled cellular growth, and body cell grow uncontrollably and also spread to the other parts. So it is not only the malignant cells which will grow and spread to the other parts, it will also draw the blood supply from the nearing blood vessel, and also it will have a characteristics of metastasis to the lung, brain, and liver. So the word cancer has been derived from Latin word, which presumably because it adheres to any part that it seizes in an obstinate manner like a crab. The WHO defines cancer as a rapid creation of abnormal cells that grow beyond their usual boundaries, which
ஒரு
can then
ಪ್ರೊಫೆಷನಲ್
invade
ಕೆಲಸ ಬಾಂಡ್
adjoining parts of the body and spread to
ಮಧ್ಯದಲ್ಲಿ ಅವ್ರು
other
ಬೇರೆ
organs. Cancer is also known as malignancy or neoplasm. How does this normal cell differ from a cancer cell? In a cancer cell, you have a uncontrolled growth of cells. They avoid apoptosis. They keep replication. They sustain their tumor nutrient supply from nearing blood vessels, and they are very insensitive to anti-growth signals. And also they have got the potential of invasion and spreading to other tissues. So before we move on, let us understand certain nomenclatures about growth. Growth means increase in size due to synthesis of tissue components. Proliferation means cell division. Differentiation means functional and structural maturity of cells. Tumor means swelling or new growth or mass. If the proliferation is controlled and reversible, then we use hypertrophy, hyperplasia, metaplasia, dysplasia. Hypertrophy is pertaining to the size. Hyperplasia is pertaining to the number. Metaplasia is the change in the cell. And if the change is distorted, we call it as dysplasia. And what is this type of proliferation are usually controlled and it is reversible. If the proliferation is progressive, purposeless, pathologic, characterized by loss of control over cell division, in addition, there is a DNA damage at growth control genes leading to the development of neoplasm, and this DNA damage happens because of carcinogens which can be a chemical one, which can be a physical one, or a infection one, which leads to DNA damage and neoplasm. So we call these characteristic features of a cancer, malignancy, or neoplasm. Coming to classification of cancer, we usually classify them in two types. One is the behavioral one, and the second one is histogenetic one. The behavioral classification, we usually classify them as benign and malignant. In histogenetic component, we classify them as epithelial, those arising from epithelial as carcinoma, like example adenocarcinoma, basal cell carcinoma, squamous cell carcinoma. Those arising from bone, cartilage, or connective tissue as sarcoma, and those arising from bone marrow as hemopoietic and lymphoid origin. As we already discussed, if the proliferation is uncontrolled and irreversible, then we call them as benign or malignant one. The benign one is usually a localized and non-invasive type. The malignant one is usually the spreading and invasive type. Coming to other difference between benign and malignant, benign is usually a slow growing one. Malignant is a fast growing one. Benign is capsulated one. Malignant is non-capsulated one. Benign is non-invasive. Malignant is invasive and infiltrative. Benign usually do not metastasize, but malignant has a feature of metastasis. Benign is usually well differentiated. Malignant is usually poorly differentiated. And benign usually we use oma as a suffix, like fibroma. Here in malignancy, we use suffixes carcinoma or sarcoma, like squamous cell carcinoma. Coming to differentiation between carcinoma and sarcoma, the origin of carcinoma is usually from epithelium, but the origin of sarcoma might be from bone, cartilage, and connective tissue. The behavior of a carcinoma is usually a malignant nature, and also sarcoma, both are malignant in nature. The commonly seen entity is a carcinoma, but relatively rare we see sarcomas. And the preferred route of metastasis for a carcinoma is through lymphatic system, and for sarcoma it is through blood. In situ phase is existing in carcinoma, but there is no in situ phase in sarcoma, and carcinoma usually affects more than 50 years and sarcoma less than 50 years. Coming to the global impact of cancer, NCDs or non-communicable disease are now responsible for the majority of global deaths, and cancer is expected to rank as the leading cause of death and the single most important barrier to increasing life expectancy in every country of the world in the 21st century. Globally, the cancer is the leading cause of death, which is contributing to about 10 million deaths during 2020. 1 out of 6 deaths is attributed to cancer. If we start ranking the cancer in pertaining to the premature mortality across the world, we see that cancer has not left any place. One in 5 men and 1 in 6 women globally developed cancer during their lifetime. 1 in 8 men and 1 in 11 women globally die from cancer. The second leading cause of death, cancer, is the second leading cause of death in developed countries and the fourth leading cause of death in developing countries. 5-year prevalence, that is the total number of people who are alive within 5 years of cancer diagnosis, is to the tune of 43.8 million. If you see the distribution of cases and deaths worldwide among both the sexes, we see that the Asian continent contributes to a major cancer incidence and also the major cancer mortality. Coming to the distribution of cases and deaths for the 10 most common cancers among both the sexes, we see that lung cancer, breast cancer, colorectal cancer, and prostate cancer, followed by stomach cancer and other cancers are seen. And coming to mortality, again lung cancer, followed by colorectal cancer, stomach cancer, liver cancer, breast cancer, and later other cancers. Cancer, we can also observe that cancer pattern follows a race or ethnicity. If we see here, whites and black, there is a different cancer pattern, and among the Indian also we see a different cancer incidence and deaths which have been reported. Coming to India, cancer is rapidly emerging as a matter of public health concern in India. The cancer incidence reported is about 70 to 90 per 1 lakh per year, and the prevalence is nearly 2.5 million cases. Second and fourth leading cause of adult deaths in urban and rural India. Coming to state-wise, we see that compared to 2017 and compared to 2018, we see a gradual increase in persons who are attending to NCD clinics across the states in our country, and also increase in number of cases being diagnosed among common cancers across the country. Coming to sex, we see that in India, in males, lung cancer followed by oropharyngeal cancer, prostate cancer, colorectal cancer, stomach cancer, esophageal cancer, liver cancer, and lymphoma cancer. But in females, we see cervix, uteri, breast cancer, followed by stomach cancer, colorectal cancer, oropharyngeal cancer, lung cancer, ovarian cancer, esophageal cancer, and liver cancer. Coming to our state, there is a equal sex distribution of incidents being reported in our case and in our state, and the annual average cases that is being reported is to the tune of 9,683. 1 in 7 males and 1 in 6 females. There is a cumulative risk of developing cancer among 0 to 7 years of age group.
I don't know if my name is getting displayed. I'm not able to find where to change the name.
5 leading sites of cancer in our state. Again, in males, lung cancer, followed by stomach cancer, followed by prostate cancer, followed by esophageal cancer, and liver cancer. In females, we see the top cancer is the breast cancer, followed by cervix uteri, followed by ovarian, followed by mouth and corpus uteri. The projected incidence of cancer for 2025, we see that there is an increase of cancer cases that is going to be reported, and this increase will be again the females. There will be a larger burden of cancers which is being reported. Coming to Bangalore, we see in 2020, 13,695 cases were reported, and among them, males contribute about 6,202 and females 7,493. How does this cancer develop? The cancer develops because of change in the gene. Change in the gene which can occur due to genetic inheritance, which can occur due to errors that occur as cells during division, or a DNA damage which is due to somatic causes, which can be due to environmental, chemical, or infection causes. The chemical carcinogens can be a mutagens or a chemical carcinogenesis and their metabolism. The physical carcinogenesis, radiation can be through ultraviolet or asbestosis, and infectious pathogens, we'll discuss it later on in detail. So what happens? The DNA change causes the normal gene involved in the normal cell growth to convert it into a oncogene. The oncogenes cannot be turned off, and result is an uncontrolled cell growth. There is a tumor suppressor gene which stops the cell growth in the normal cell divisions. But in cancer cell, the DNA changes inactivates this tumor suppressor gene, which will lead to abnormal cell growth. In addition, the tumor microenvironment, which constitutes the immune cells, fibroblasts, molecules, blood vessels, this cancer cell will alter this tumor microenvironment, which determines how the cancer spreads and grows. In addition, the cancer cell will avoid the immune detection system and which also leads to an evading nature of the cancer cells. As the age advances, the genetic changes which will be inherited, which accumulate slowly, leading to a higher risk of cancer in later life. So that's how we see that cancer arises because of a DNA mutation, which can be due to heredity or due to carcinogens or due to errors during DNA duplication, which leads to an uncontrolled proliferation. And this is mainly triggered by three genes: proto-oncogenes, tumor suppressor genes, DNA repair genes, which fail to do their function. The proto-oncogenes fail to check the normal cell growth and division. Tumor suppressor gene, controlling of cell growth and division, and DNA repair genes, which fixes the DNA damage, fail in cancer, because of which there is a uncontrolled proliferation. What triggers this? What triggers this? What triggers these things are a set of risk factors. The set of risk factors are categorized as age, or sex, or heredity, lifestyle, cancer-causing substances, chronic inflammation, hormones, immunosuppression, infectious agents, radiation, sunlight, which can again can be categorized into two components. One is modifiable and the another one is non-modifiable. So the modifiable factors predominate, which include the tobacco use, alcohol consumption, unhealthy diet, physical inactivity, air pollution. The non-modifiable factors include age, gender, a familial predisposition, or genetic predisposition. As we see, as the age increases, the chances of being diagnosis of cancer also increases. The median age of cancer diagnosis is usually 66 years. This means that half of the cancer cases occur in people below this age and half in people above this age. A similar pattern is also seen in many common cancer types. But we should never forget that the cancer can be diagnosed at any age. For example, bone cancer, which is diagnosed usually within 20 years. And brain and other nervous system cancers, which are detected in 12% of children and adolescents. The gender distribution of cancer, as reported in US, also we see a different pattern. As we see in Indian scenario, lung cancer predominates, and in women, breast cancer predominates. In US, we see that the prostate cancer predominates, and breast cancer again in women predominates. Lifestyle is the most important factor which determines, which include smoking, obesity, excessive sun exposure, unprotected sex, poor diet, sedentary lifestyle, or alcohol consumption. Alcohol consumption is associated from leading to cancers in the breast in women, colon and rectum, liver, esophagus, throat, mouth, voice box, and around 3.5% of cancer deaths in United States are accounted due to alcohol consumption. Diet, here the results which are obtained, some results are inconclusive, some results are confirmatory in animal studies, but the facts which is laid down is the diet having acrylamide, which is found in tobacco smoke and some foods, produces, which can be produced when certain vegetables like potatoes are heated to high temperature, is has been found as one of the carcinogens. Antioxidants are chemicals which are block the activity of other chemicals, are known as free radicals, are found in animal studies that they may damage cells. But in human studies, the results are inconclusive. Artificial sweeteners, the role is again yet unclear. In a charred meat, certain chemicals like HCS and PAH are formed when muscle meat, including beef, pork, fish, and poultry, is cooked using high temperatures. Methods have shown leading to cancer. Foods which are beneficial in preventing cancer are calcium, fluoride, tea, which as it contains polyphenol compounds, particularly catechins, which are antioxidants again, vitamin D, which helps the body use calcium and phosphorus to make strong bones and teeth, and has reduced the risk of colorectal cancer. Cruciferous vegetables, which contain chemicals known as glucosinolates, which break down into several compounds that are being studied for possible anti-cancer effects. But all these beneficial entities, again in humans, the studies have been inconclusive. Coming to overweight and obesity, cancers in an obese person have been found in thyroid, breast, liver, gallbladder, upper stomach, pancreas, colon and rectum, meningiomas, adenocarcinomas of esophagus, multiple myeloma, kidney, endometrium, and ovary. Tobacco, in fact, leaves no particular organ. In fact, it attacks anywhere in your body. Smoking tobacco products, including cigarettes and cigars, causes almost 9 of every 10 cases of lung cancer. Chronic inflammation is another risk factor. Usually, inflammation is a normal physiological response that occurs by an injured tissue to heal. But in chronic inflammation, this inflammatory process may begin even if there is no injury, and it does not end when it should. When the inflammation continues, is not always known. Why this inflammation continues is always not known, and chronic inflammatory bowel diseases such as ulcerative colitis, Crohn's disease have an increased risk of colon cancer. Hormones like estrogen and progesterone are found to be associated with breast cancer, prostate cancer, hormones like testosterone, androgen. Hormones of estrogen and progesterone again have been commonly associated with ovarian cancers. Growth hormone are associated with bone cancer, thyroid hormones with thyroid cancers. Coming to infectious diseases, we have a Epstein-Barr virus, which is a type of herpes virus, causes mononucleosis as well as a certain types of lymphoma and cancers of nose and throat. Hepatitis B virus and hepatitis C virus can cause liver cancer. Human immunodeficiency virus does not cause cancer itself, but due to weakening of the immune systems and making it vulnerable to infectious agents, will have a predisposition, especially for Kaposi's sarcoma, lymphoma, and cancers of cervix, anus, lung, liver, and throat. Human papilloma virus have increased potential for cervical cancers. Human T-cell leukemia or lymphoma virus type for non-Hodgkin's lymphoma or T-cell leukemia or lymphomas. Kaposi's sarcoma are associated with herpes virus. Merkel cell polymovirus, Merkel cell carcinoma, Helicobacter pylori are by a type of are associated with a type of stomach cancer and a type of lymphoma in the stomach. Opisthochisis Urinary parasitic flatworm, which is found in Southeast Asia, can cause cancer of the bile ducts. Schistosoma hematobium, a parasitic flatworm which lives in certain types of freshwater snails found in Africa and Middle East, can cause bladder cancer. Now, we have, as we have known that there are so many risk factors which can cause the increase in cancer risk, but nearly half of these deaths will be attributed to the cancer caused by potential modifiable risk factors like tobacco smoking, overweight, alcohol, exposure to sunlight, poor diet, infections, and physical inactivity. So all these factors can be modified, are attributed to the modifiable risk factors. So the true prevention is not waiting for bad things to happen. It is preventing things from happening in the first place. So as we see the word cancer, the cancer has can into it, the positive word into it. So together we can beat it. So with this brief note,
Microsoft
I
links.
thank.
Alumina has some importance. Shashidharan Manipal all link ఇవ్వకపోతే ఎవరికి ఇస్తాడు? క్యాన్సర్ ట్రాన్స్ అమ్మా ఇది ఇవాళ
Thank you, sir, for your exemplary talk. Now the forum is open for discussion. Participants can text your queries on chat box, which will be addressed. So there is a question. Diabetes as a risk factor for cancer. Any relationship?
Hello. Hello.
Yeah, Anil.
Sir. Am I audible, sir?
Yes, yes.
Sir. Hello.
Sir, yes, sir, you're audible, sir. Please,
Yes,
go ahead, sir.
Yeah. What is the question, sir?
Sir, diabetes as a risk factor for cancer, sir. Is there any relationship? Is a question posted by our audience.
As far as my knowledge goes, non-communicable diseases, especially unless and until it is associated with decrease in immunity response like HIV, I don't think so. If the diabetes is linking to a decrease in the immune response, then prone for infections, and that can cause as a viscous cycle leading to cancer. Hello.
Yeah, Anil.
Sir.
Please go ahead. Yeah.
Answer. That can lead to going ahead with the cancer, but most of the carcinogens which are listed are either related to environmental carcinogens or toxic related chemical carcinogens and infectious related carcinogens, which will lead to the DNA gene change in the gene cancer causing gene. So that is the potential risk factor.
Can I add a few points, Dr. Anil?
Answer. Answer.
We have worked with respect to the cancer and diabetes, in which we try to link the fluoride with respect to the cancer. So here, with respect to the cancer, what will happen, the biochemical basis, what we have observed in our study, the Kolar population, the local population, is highly exposed to the fluoride concentration. The fluoride is supposed to inhibit the enzyme enolase. When it inhibits the enzyme enolase, what will happen? As we all know, the effect of the product that we have studied in the enzymology, the subsequent, the prior substrates, the concentration of these substrates gets increased. Ultimately, in case of glycolysis, the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, the concentration is going to get increased. So this in turn will lead to increased concentration of the triacyl glycerols. These triacyl glycerols are trapped by the LDL. And this LDL, in turn, what will happen? It undergoes oxidation. And this oxidized LDL is going to affect the cell membrane. So when it affects the cell membrane, there is an impact that the molecular basis that we have observed with respect to this oxidized LDL. So the cell membrane, when it gets weakened, then what will happen? There is a chance for the carcinogens to enter into the cell and leading to the carcinoma. This is the observation that we have done. We have done one ICMR project with respect to linking the lipids, the diabetes mellitus, the fluoride. So with this is the observation that we have observed. Further, we have observed that these population, what we have observed, so the glycated hemoglobin, the concentration were very high. It was around 13.3. The mean observation that we have observed was around 13.3. And this uncontrolled and the long-standing diabetes may be one of the reason for these people, as Dr. Anil has rightly said, the immunocompromised condition and the decreased immunity might have led for increased exposure or the increased chance of getting the virus-linked cancers and various other molecules to enter into the cell to cause mutations in the DNA and ultimately leading to the cancer. This is what we have observed in our study and our experience. Over to Dr. Anil.
Dr. Anil?
Sir.
Ravi Shankar here. The question I want to know, is there any direct relationship of diabetes with cancer based on for example, consistently high increased blood sugar level, does it causes any relation with cancer? That's what I want to know.
Sir, I have not come across any study with a direct correlation. Maybe I think I'll get back to you, sir, by going ahead with some more literature.
Okay. Thank you, Anil. Okay. Very nice presentation. Thank you very much.
Thank you, sir.
Thank you, Dr. Anil.
Thank you, sir.
Dr. Soumya.
Thank you, sir. Now I request Dr. Akila ma'am to present on emerging biological mechanisms linking obesity to cancer. To say a few lines about Madam. Madam is Professor, Department of Biochemistry and Convener, Department of Medical Genetics, JSS Medical College, JSS Academy of Higher Education and Research. Ma'am has played a vital role in availing the Department of Science and Technology funds for improvement of science and technology, infrastructure in universities, award to the department in 2012 for a period of 5 years with a grant amount of 80 lakhs. Ma'am has been successful in obtaining funds from government agencies like ICMR, Vision Group on Science and Technology, and DST. Madam's current areas of research include molecular biology and genetics of rare disorders. And ma'am is currently working on a multicentric project, National Registry for Rare and Other Inherited Disorders, and has been awarded the prestigious Professor Y.T. Tatachari Endowment Prize for the Best Research Scholar at the Graduate Ceremony 2015 at JSS Medical College, and Appreciation Award for the research activities carried out in the year 2019 by the Research Division, JSS Academy of Higher Education and Research. Women Achiever Award 2021 by JSS Medical College on the occasion of Women's Day, apart from the Best Oral and Poster Presentation Awards in the national and state conferences. Ma'am has organized 6 national hands-on workshops in molecular methods in disease management and has trained 100 delegates to date, and has also been invited as resource faculty by various institutes. Ma'am has established and developed the Department of Medical Genetics at JSS Medical College, which has facilities for newborn screening for treatable metabolic disorders, maternal serum screening for the detection of aneuploidy and chromosomal disorders, and clinical cytogenetics having facilities for karyotyping and fluorescent in situ hybridization. MAM has initiated BSc Medical Genetics and MSc Medical Genetics and various genomic programs, for which the syllabus has been designed according to choice-based credit system, that is CBCS. MAM has nearly 50 publications to her credit and has published in various indexed national and international journals. MAM is guiding 7 PhD scholars on various research topics. MAM has also taken keen interest in employing innovative methods in teaching, learning, and evaluation, and have initiated problem-based learning, objective structured practical examination, experiential learning, and model making exercises for the medical students. I welcome you, ma'am. Over to you, ma'am.
Yeah, thank you very much for the kind introduction. And at the outset, I would like to thank the organizing committee for giving me this opportunity to present a little bit of our knowledge and experience what we have with respect to the metabolic disorders and cancer. So I'll just start sharing my screen. Is the slides visible?
Yes, ma'am.
So I'll be actually very much thankful to Dr. Anil, who has set up the platform for the discussion, and he has given an overview of all the risk factors for cancer. But I'll be concentrating more on the link between the obesity and cancer. So this would be the content I'll be discussing about: a brief introduction and then the epidemiological link, evidences which have shown link between obesity and cancer. And what are the reliable indicators for obesity? Is it the BMI, or we have to look for some other indicators for obesity? And then we'll discuss some of the pathways, biological mechanisms which link the overweight or obesity to cancer. And finally, the take-home message. So if we look at the world map of cancer, this is the latest incidence, age standardized incidence worldwide for all cancers in both sexes and all ages. So we can see that no region is spared. Every region has been affected by this disorder, and it's a major global concern for everyone, and we have to find the researchers everywhere are working to find best therapeutic targets for handling this particular disorder. So this is the latest statistical map which shows the age standardized incidence of cancers worldwide. So most common cancers in females and males, if you see it as in females, we see the breast cancer, which is most common, followed by the colon, lung, and cervix. And in case of males, it is a lung cancer, which tops the chart, and then the prostate, colon, stomach, and the liver cancers and other cancers. So there are various factors which can lead to cancers, which was told even by Dr. Anil. So it can be the biomedical factors, the lifestyle factors, the environmental factors. So biomedical factors like the genetic susceptibilities, hormonal factors in case of females, environmental factors like sunlight, radiation, occupational exposure, pollution. Coming to lifestyle factors like smoking, alcohol consumptions, and what we are interested today in this talk is the physical inactivity and obesity, which also is a major risk factor for the development of cancer. The incidence of obesity, if we see worldwide, even you can see that males and females, the obesity is also quite predominant worldwide, and again, no region is kind of spared by this condition as well. So there is a varied distribution, some areas being widely affected by this condition, and it all depends upon various causes why obesity is also quite widespread. So why we are so much concerned about obesity? because this obesity can lead to various disorders like dyslipidemia, type 2 diabetes, hypertension, liver disease, reproductive disorders, heart disease, mood disorders, as well as it is also a risk factor for cancer. It has been recently, people have started working on finding the link between the obesity and cancer, why there has been shown increased risk of cancer in obese individuals. So what are the different cancers which the obesity is leading to is various cancers. You know, there is not one in particular. Different organs are affected by, could be affected in the obese individuals. It could be the colon, other gastrointestinal system, the ovarian, breast, liver, endometrium. So various cancers may be due to the condition of obesity, and this obesity kind of, you can see the incidence in males, it's about 14% worldwide, and females about 20% worldwide. This is not just a hypothetical link which has been shown, but there has been statistical evidences which has linked the obesity to cancer. So there are certain studies which have shown increased relative risk of obesity being linked to cancer. And you can see, for example, very high relative risk, more than 3, has been shown linking obesity with endometrial carcinoma and esophageal carcinoma. Moderate relative risk has been shown in renal adenocarcinoma, hepatocellular carcinoma, pancreatic, gastric, multiple myeloma, and meningioma. And still but still it's a significant risk in case of colorectal carcinoma, post-menopausal breast cancer, gallbladder, ovarian, and thyroid cancer. And what we are working mainly is on the colorectal and the post-menopausal breast cancer. So there are these statistically significant link which has been shown, has been linked obesity with cancer in at least 13 anatomical sites. And these sites are mainly predominantly the digestive organs, the cancers of the hormone sensitive in women. And also there is an interesting epidemiologic aspect in finding that the excessive body weight and comorbidities have been increased even in cancer survivors. So those who have been treated and survived of this cancer, there is an increased risk of obesity seen even in these cancer survivors. So the schematic diagram which shows the overview of these links. Okay, so it is we have the adipose tissue, which is a white adipose tissue as well as the brown adipose tissue, but this white adipose tissue is the one which is more riskier for the development of cancer. So this pathological expansion of this white adipose tissue in various abnormal sites like the intrahepatic, intra-abdominal, and intramyocellular sites can lead to metabolic, inflammatory, or immunological alterations, which can affect the DNA repair, the mutation rate. There are some epigenetic changes which can happen. All this together will lead to the malignant transformation and the progression of cancer. So not only these epidemiological studies, we also have some data about a small study which we had done with respect to a retrospective study. We had collected data for the past 5 years of all the breast diseases which have been treated in our hospitals. About 420 breast diseases were treated in our hospital. In that, a few of them we had to eliminate because they were benign conditions, and some we did not have any data of the patients. Finally, 335 breast cancer cases were shortlisted. Among these also, there were some conditions, some patients in whom we did not have the complete data, so we had to eliminate those. Finally, about 250 cases which had complete data was included in the study. So when we categorize these patients based on their BMI, we can see that most of these patients, about 173 patients out of the total number what we had screened, about 173 patients were belonging to the overweight and obese category. Okay, so and among them we found that about 120 of them, you can see here, about 120 of them were of postmenopausal age. So not discussing all the results here, but just to highlight here that we also did find the overweight being linked to cancer, and especially in case of breast cancer, since the literature says that it is postmenopausal obesity which is more risky for the breast cancer. So even our data showed that the postmenopausal women were having overweight were at risk for the breast cancer. And their follow-up also, we found that these patients with the postmenopausal women, especially having increased increased BMI, they had, we found more incidence or increased incidence of death as well as recurrence in them. Okay, so this is a small study which we have done. So there are multiple parameters which we have analyzed in this, but this is something which I wanted to highlight here. So is it only the BMI, or is it only the one-time weight which is important of a person, or should we monitor the weight and see if there is any change in the BMI which is important for the development of cancer? Okay, so that was again a paper which has been published some few years back, which shows that it is not the one-time BMI which is important, it is the gain of weight which is important. So adult weight gain, which they say is a better indicator than BMI, because it helps us to know the dynamic nature of the obesity or the adipose tissues. So they have done a meta-analysis combining, several studies have been used in this meta-analysis, and the relative risk of these studies have been shown here. So it has, we can see here, it is the relative risk increases when there is 5 kgs gain in the weight. Okay, so there is an increase of relative risk when there is 5 kg gain. So it is like not the one-time BMI which would be important, but the gain of weight over the years would be important for us to say whether there is a risk of development of cancer. And not only this gain of weight, the intentional weight loss has been shown to lower the risk of cancer. Okay, so particularly in obesity associated cancer in women, it has been shown that the intentional weight loss. So this intentional weight loss can be due anything when the person intends to lose weight by either physical exercise or surgical route or medication, but there is some intentional weight loss which has also shown to lower the risk of cancer. Now, coming to the indicators of obesity, do we choose BMI as a good indicator? There are several studies which say no, actually. So BMI is not a good marker for obesity because it cannot differentiate between the adipose tissue and the lean mass. Okay, so that is one of the major disadvantages. And BMI is inaccurate to evaluate the obesity in elderly subjects, individuals with extreme height, very muscular individuals, and so these are the some of the individuals in which we will not be able to estimate the BMI accurately. So BMI is not as such chosen as a good indicator for obesity, and it will also underestimate the visceral obesity because visceral obesity is the one which is important for linking our risk to cancer. So BMI does not actually estimate the visceral obesity properly. And sometimes women tend to present higher fat percentage than men if BMI is taken as an indicator. So it is, there are several reasons why BMI may not be the best indicator for obesity. So what next? What other indicators can be chosen? So waist circumference has been shown to be a good indicator because it is it strongly associates with the visceral fat. Yeah, it has been shown as a better indicator for cancer risk. Waist-hip ratio, that is also strongly associated with the visceral fat. So again, that could be taken as a indicator for obesity. Fat distribution, as I told you, it is a visceral adipose tissue which we have to consider rather than the subcutaneous adipose tissue. So how do we estimate the visceral adipose tissue? For that, it is only the CT scan, which is a standard method for quantification. But it is not feasible. Doing CT for all the population-based studies would not be feasible. So instead of CT scan, the waist circumference or waist hip ratio could be a surrogate in markers for or measurements for estimating the visceral obesity in the population-based studies. And of late, NAF-LD also has been shown to be a good indicator, but again, it has to either ultrasound or some other things have to be done. But the best one would be the waist circumference and the waist hip ratio. So there is a small study which we had done on the healthy obese, healthy individuals, and characterize them as obese and non-obese, but then we didn't find good correlation with that. Then we classified these individuals as insulin resistant and insulin sensitive based on the HOMA-IR index. So based on that, we found a good correlation with the neck circumference of the individuals, and classifying this individuals with based on the IR and IS, that is insulin resistant and insulin sensitivity. We found that we can eliminate the bias which can be due to the subcutaneous fat. Okay, so here you can see that the subcutaneous fat is not significantly different between the IR and IS, but the visceral fat index, we can see it is significantly different. So IR can be a representative marker for the visceral fat index. So this classification based on IR and IS can be used to actually classify the individuals based on their obese parameters. Also correlated whether all these parameters, the Pearson's correlation was done with BMI and HOMA-IR, and here also we found that the subcutaneous fat actually was showing a correlation with the BMI, but when you take the HOMA-IR, the subcutaneous fat is not showing a correlation with HOMA-IR, but instead visceral fat is the one which is showing a good correlation with the HOMA-IR. So this indicates that HOMA-IR can be used with more reliable feature when we want to work on obesity and risk for certain disorders like diabetes, hypertension, or cardiovascular disease, or with case of cancers. Instead of BMI, it could be these other markers like waist hip ratio, circumference of HOMA-IR, which classify or stratify the patients. Now, coming to the biological mechanisms linking overweight to obesity, there are several mechanisms, several pathways. Okay, the most important ones like hyperinsulinemia and insulin resistance, the sex hormone biosynthesis pathway, subclinical or chronic low-grade inflammation, alterations in the adipokine pathophysiology, and some of the factors which are deriving from the ectopic fat deposition, the microenvironment surrounding the cancer cells, disruption of the circadian rhythm, dietary nutrients, as well as altered intestinal microbiome. So there's several reasons, several pathways. It's not one pathway which is linking obesity to cancer. There's several pathways linking obesity to cancer. So briefly about the important pathways. This is one of the pathways which has been very well studied, actually, that is insulin growth factor 1 axis and insulin resistance. There was a question on how diabetes is linked to cancer. So we can use this pathway to show the link of diabetes with cancer as well. So this is a recent screenshot of a recent publication which has so here we can see that several metabolic factors like metabolic syndrome, obesity, diabetes mellitus, acromegaly, they're all risk factors for cancer because they increase the levels of insulin growth factors.
ಆಮೇಲೆ ಪ್ರೂಫಿಗೆ ಅದೇ ಎಕ್ಸೆಲ್ ಶೀಟ್ ಹಾಕಿರಲ್ಲ ಇಟ್ಕೊಡಿ okay ನಾನು ಮಾಡಿದ ಕರೆಕ್ಟ್
one is increased in all these metabolic syndromes. So this insulin growth factor 1 is also released by the white adipose tissue. So whenever there is increased obesity or increased deposition of the white adipose tissue, there is increased secretion of this insulin growth factor. This dysregulates the pancreatic cells, and here it will increase the secretion of insulin because there is dysregulation. It does not sense the glucose levels, and though the glucose levels are high. The regulation does not happen properly. So there's dysregulation, increasing the insulin levels, and this hyperinsulinemia further has risk for these metabolic disorders. Also, this insulin has it increases the proliferation and apoptosis of the cells and increases the size of the tumor. What it does is that it activates these pathways, that is PI3K, AKT, mTOR pathways, the RAS, RAF, and MEK pathways, JAK-STAT pathways, inhibits the AMPK pathway, okay, and it also has it shows certain actions on the GLUT receptors, which it increases the GLUT receptors on the surface, which increases the uptake of glucose. And as well as all the enzymes, key enzymes of glycolysis are also activated, and there is increased aerobic glycolysis which happens here. And this increased glycolysis, which is termed as Warburg's metabolic phenotype. So that is something which is commonly seen in the cancer cells. So this increased glycolysis further proliferates and facilitates the survival and invasion of the cells. So this is how insulin and increased hyperinsulinemia and increased glucose levels are linked to cancer. And we have seen that not only this link has been shown, it has been several publications have been there now where it has been shown that oral anti-diabetic drug metformin, which is used actually for the treatment of diabetes mellitus, but this drug also shows anti-cancer activities. Several studies, several researchers have been working on repurposing of this metformin into the treatment of cancer. So this metformin along in combination with insulin, moderate physical activity and a good Mediterranean diet would help in reducing the incidence of cancer. Not only this metformin, but there are current therapeutic strategies which have been used to target the aerobic glycolysis, because it's the aerobic glycolysis which has been increased in this cancer cells, that is Warburg's effect. So several therapeutic targets we can see here as per the latest publication, which has effects on each one of these pathways, which I mentioned has been increased in the cancer conditions. So there is, there are agents which can suppress the PI3K, AKT pathway, AKT mTOR pathway, suppresses the phosphorylation of ERK1 and 2. And there are several microRNAs and long non-coding RNA, which shows inhibition of this IGF-1 receptor, and also the STAT signaling also it suppresses. And this is a long non-coding RNA, which has which shows its action on this PI3K, AKT, and mTOR pathway. So that is one of the pathways which has been shown to link obesity with cancer. Next, we have the steroid hormone, sex hormone biosynthesis pathway. Sex hormones, as such, we know that it is the gonadal cells which are the ones which are synthesizing the sex hormones. So in adrenal glands, the androstenedione and testosterone, which is released, it in the ovary, it gets converted to estradiol and estrone. And because there is aromatization which takes place with the help of the enzyme aromatase, this estrogen then comes and binds with the estrogen receptor, and then this signal transduction which takes place, which leads to proliferation of the cells. Now, this aromatase enzyme, it's not only that it's present in the breast tissues, the ovaries, but it's also seen in other peripheral tissues, like in case of adipose tissues. So in case of obesity, this levels of these aromatase enzyme increases, and more and more of testosterone gets converted to estrogen, and this leads to whenever there is estrogen receptor levels increased in the breast cells, then the signal transduction takes place after the binding of estrogen to this estrogen receptors, leading to proliferation of the cells. Now, what happens in the postmenopausal woman? The ovaries are not functioning now, so this adrenals glands, what androstenedione and testosterone is released, they are converted to estrogen by the aromatase, which is there in the adipose tissues. So that's why we see more of the cancer seen in obese postmenopausal women. Okay, so this aromatase, because there is dysregulation here in case of the premenopausal woman, the adipose tissue aromatase will not be active, and it will be regulated to its tissue itself. But in case of postmenopausal woman, this dysregulation of this adipose tissue aromatase, and it can convert more and more of androgens to estrogen, and there is increased proliferation. So there are several therapeutic agents which have been used to target each reaction in this pathway. So we can see that there is estrogen receptor modulators which have been used. It is mainly tamoxifen, which is given to ER/PR positive breast cancers. So it's mainly treated with tamoxifen. This tamoxifen is estrogen receptor modulator. So that inhibits this binding of estrogen to the estrogen receptors, and then that's how it decreases the proliferation. So there are several agents which can be used, so aromatase inhibitors or receptor destabilizers, which can be used to target these pathways and inhibit the formation of estrogen here. So there can be anti-estrogens which are used, anti-androgens which are used, and there have been 17-20 lyase inhibitors which have been used to target the sex hormone biosynthesis pathway. So in this regards, we had done a study on the cell lines, that is MCF7 breast cancer cell lines. MCF7 is a ER positive cell line. So these cells were plated, and after it was like 80% confluent, it was treated with the tamoxifen, which is a known drug to treat the ER positive cells. And the tamoxifen, as you can see, it is since it's a known drug, we have seen a good inhibition with the tamoxifen. So there's about 80% inhibition at the highest concentration, and there's dose-dependent response. We can see how much inhibition was there at 24, 48, and 72 hours. Now, these same cells, MCF7 cells, when they were exposed to leptin, so you can see that these cells increase the proliferation. So there is the bars you can see on the negative side. So it shows that there is increased proliferation because it is an obese condition which has been mimicked here. So there's increased proliferation which has seen at 24 hours, 48 hours, and 72 hours as well. But at 72 hours, because the concentration of leptin is very less here, so there is a little bit of inhibition which has been shown. But now, so this indicates that the obese conditions is favorable for the cells to proliferate. Okay. Now we in the when the cells had an increased concentration of leptin with a dosage of 10, 20, and 100 nanogram per ml. So 10 mimics the normal leptin levels, and 100 is the obese leptin levels. So when these cells which were exposed to already leptin were treated with tamoxifen, so we can see that the percentage inhibition is less when compared to this. So here we have 80% inhibition, but here there is only 40% inhibition. So it says that not only that obese conditions are helping the cells to proliferate, but also it will decrease the efficacy of tamoxifen as well. Okay, so so not only it's obese patients are at risk for cancer, but the anti-cancerous agents also might not be effective for the obese individuals. So this study, we are further continuing this, and there are several gene expressions analysis which are pending from this study. So that data I didn't have here. So this was a preliminary work which we had done. So it gives us a good platform to further proceed with the gene expression studies and other assays which further proves that the obese conditions would be risky for even the treatment as well as the occurrence of cancer. So cancer, as obesity, we know it's a chronic low-grade inflammation, and this also is one of the reasons why the chronic low-grade inflammation and oxidative stress is an increased risk factor for cancer. So in case of obesity, there is a chronic inflammation, adipokine imbalance is there, oxidative stress is also seen, which leads to increased secretion of these pro-inflammatory cytokines, adipokine imbalance, which you can see the increased leptin and the decreased adiponectin levels, and also the ROS and the nitric oxide reactive species also has been increased. So which converts this normal epithelium to adenoma and carcinoma. So you can see here this in case of chronic inflammation, this inflammatory cells which have increased ROS and the reactive or nitrogen species, as well as increased secretion of the cytokines. They can induce certain mutations, epigenetic changes, which can transform the cells to cancerous cells. They activate these pathways like NFK beta, STAT3 pathways, MAP kinase pathways, and AKT pathways, which further will proliferate the cells, improve their survival. These cells will have the invasive properties and also has properties to synthesize their own blood circulations, angiogenesis happens, which will help in the tumor promotion and progression. Okay, so this is also another pathway wherein how the obesity is linked with cancer, because both are kind of low-grade inflammatory conditions. Of course, crosstalks takes place between this cancer and the tumoral adipocytes. So you can see here, these are the mature adipocytes. These mature adipocytes will secrete the chemokines and cytokines, which helps in homing the tumor cells in the adipose tissue as such. And once these tumor cells come in contact with the mature adipocytes, these mature adipocytes, they get transformed to cancer associated adipocytes. Okay, so you can see this transformation which has occurred. This mature adipocytes get transformed to cancer associated adipocytes, and these cancer associated adipocytes, you can see they release the exosomes, they release the solid factors, free fatty acids. They also release this extracellular matrix factors, which help in the invasion. It can help these tumor cells to invade into the blood circulation and disseminate to the distant organs, leading to metastasis. Okay, so this kind of crosstalk between the adipocytes and the tumor cells is also important, is one of the factors which will help in the invasion and dissemination of these cancer tumor cells. Also, there is crosstalk between the adiponectin and the insulin growth factor 1 receptors. So in the breast cancer cells, so you can see this is the adiponectin molecule, and the globular domain of this adiponectin, it's a full adiponectin molecule. This is only the globular domain which is seen here. So in case of adipocytes, there is increased the release of this adiponectins. So this adiponectin comes and binds to this adiponectin receptor. Once this binding happens here, there's crosstalk between the membrane and estrogen receptors and the insulin growth factor 1 receptor. So there's a crosstalk which happens, which increases the release of the insulin receptor substrates, and also the ER alpha, which activates the MAP kinase, and then that will increase the transcription of genes, increase the cell proliferation, migration, and reduces the apoptosis. So this is how there is progression of cancer because of the crosstalks between the adiponectin and the IGFR receptors. So this is also shown mainly with respect to the breast cancer, but wherever the tumor cells comes in contact with the adipocytes, this crosstalks can happen. Also, as I mentioned earlier, also the tumors, for them to survive, it requires the circulation, because with the circulation they get the nutrients. So there's an angiogenic switch which will be turned on. Suppose if there's these tumors have to grow, and even if there is a metastasis which has to happen. So angiogenesis is something which is important for the growth of these tumors. So there is a kind of a switch which is switched on because of the presence of this pro-angiogenic factors. So we can see this pro-angiogenic factors like angiopoietin, VGF, leptin, TNF alpha and TGF. These are all the factors which can switch on this angiogenic switch. So you can see that leptin is also one of the factors which is again released by the adipocytes. So in case of obesity, the switch will be on much faster and leads to angiogenesis and the growth of the tumor. So the switch usually is controlled by the balance between the pro and the anti-angiogenic factors. When this balance is disrupted, then there is always vascularization of the tumors, which can happen. So neovascularization, it supplies essential nutrients and oxygen for the growing tumor, and it promotes the tumor's survival and metastasis. So there can be these anti-angiogenic factors, which have been again used for the controlling of this tumor growth. And so it's like a multi-pronged approach. So it's not that one pathway can be targeted. There has to be multiple pathways which need to be targeted to tackle this tumor cells. And epithelial to mesenchymal transition, this is again one of the important mechanisms which can lead to invasion and metastasis. So when these cells, epithelial cells become the mesenchymal cells, so when this transition is happening, it can invade and get transported to a different site where it again becomes there is again transition from the mesenchyme to epithelial, and it gets deposited in those tissues, and there is further proliferation which happens. So you can see here the adipocytes which are present around the tumor cells. These adipocytes can induce expression of the mesenchymal markers and promote the invasion and migration of the breast cancer cells, and help these to go and get deposited in another site, leading to metastasis, which is commonly seen in the breast cancers. So this again, epithelial to mesenchymal transition can be studied further for targeting this pathways or this transition. This transition can be again targeted, and the cancers can be prevented from being metastasis to different site. So again, this is an interesting mechanism. It's a disruption of the circadian rhythm. Here, this is one study which we have started working on actually. Here we have the central clock and the peripheral clocks. So the central clock, which is there in the hypothalamus, it receives the light signals, and it maintains the circadian rhythm. There has been certain data which shows that the night shift workers, because of the disruption of the circadian rhythm, the night shift workers having increased risk for the development of cancer, because melatonin, which is actually secreted in the night when we are asleep. So this melatonin would not be, it has got an anti-cancer activity. So in these night shift workers, the melatonin is not secreted to the level it has to get secreted. So this, because of they do not have this melatonin level, so there is increased risk of cancer in the night shift workers. We have initiated the study on not the central clock, but the peripheral clock, wherein we would like to see how the circadian rhythm genes in the peripheral tissue are different in the cancer as well as the normal individuals. Okay, so this is a study which we have just initiated, and some cell line data what we have right now gives us a promising results, which says that there should be synchronization of the cells before the chemotherapeutic agents are given to the patients. So synchronization of the cells can be brought in by several mechanisms in the in vitro assays. So one of the mechanisms what we are using is the serum shock mechanism. So wherein the cells are starved overnight, and then next day the cells are grown in a high fetal bovine serum. It's 50% fetal bovine serum. And then later on, these cells would be treated with tamoxifen, and we see that once the cells are synchronized, how the tamoxifen, efficacy of tamoxifen can be increased or not. So this disruption of the circadian rhythm, it has got various effects, because it is closely linked with the cell cycle. It is closely linked with the DNA repair. It is closely linked with apoptosis, as well as other metabolic pathways like glycolysis and TCA cycle. So there is a close link between the circadian rhythm and all these pathways, and these pathways we very well know any kind of changes happening in this pathway. There is disruption of the regulation, and the cells, normal cells, can get transformed to cancerous cells. So all these mechanisms are the ones which are linking the obesity to cancer. Again, recently there has been a lot of interest in studying the intestinal microbiome, and again how this intestinal microbiome is related to various disorders, not only cancer or obesity, various other metabolic disorders, even cardiovascular disorders. All these disorders, it is like it is said that we are what we eat. So maintaining a healthy intestinal microbiome is very important, and that's the reason why all these probiotics also have come up, which helps us in maintaining the healthy intestinal microbiome. So we can see here the intestinal microbiome here. When there is alteration of this gut microbiota, there could be which is generally noticed in case of obese conditions. So what happens is that it leads to a thinner mucus layer. So this mucus layer becomes thinner, and there is disruption of the tight junction protein and distribution of the localization. And you can when this tight junction is disrupted, there is leakage of this LPS into the walls of the intestine. This leads to activation of the toll-like receptors, and also NFK beta has been activated, which leads to low-grade inflammation. So this low-grade inflammation, which we have discussed earlier also, which has a lot of links with obesity, diabetes, cancer, as well as cardiovascular disorders. So that way, this is also one area which we can work on to see if this intestinal microbiome has any association with cancer. So considering all this, we know that the obesity has been linked with cancer, but now there are certain studies which have shown that there is no link of obesity with cancer. Okay, so probably those this is called as obesity paradox. This can be probably because BMI was used as a measure for the obesity. So we have already discussed that BMI may not be a good indicator, and it's not the one-time BMI. It is the weight gain which has to be seen over a period of time, which is a better marker for the risk of cancer. And also maybe these studies have limitations, including inadequate adjustments for confounding factors like the selection, stratification, and detection biases would be there in these studies. Confounders such as age, smoking, and physical activity also has to be taken into consideration when we are doing these studies. And the reverse causality which have been reported in certain studies may be because the weight at diagnosis would be taken into consideration, and the weight at diagnosis may be less because of cancer cachexia. So these are some of the factors which we have to take care while doing these kind of studies when we are trying to link obesity with cancer. Now, where are we in 2020 where, in terms of cancer prevention through weight control? So this is again a recent publication which came across in the British Journal of Cancer. So it still says that there is clear gaps which remain in our understanding of how changes in the body fat and levels of physical activity are mechanistically linked to cancer and the magnitude of their impact on cancer risk. So still there is a lot of gap. Why I'm stressing on this is because there is a lot of scope to work on this, and we can can start working on these areas, and we can have a good number of data and publications when you come across good results which you can generate from your lab. But you have to take care about the confounding factors and the proper measurements what you are using for your studies to stratify the patients. Finally, the take-home message here, I would like to say that there is sufficient evidence for a strong connection between obesity-driven chronic inflammation, insulin resistance, adipokines, and the altered microbiome and cancer. But further research in basic and translational research is essential to delineate the this role of adipocytokines and their interplay in obesity-related cancer pathogenesis. And the prospectives and longitudinal studies are expected to determine a broad spectrum of obesity-related biomarkers and evaluate their clinical utility in cancer prognosis and monitoring. And a significant percentage of cancer cases can be preventable by lifestyle changes like quitting smoking, maintaining a healthy weight, following a healthy diet, and increasing the physical exercise and decreasing the alcohol intake. So a lot of things can be prevented by lifestyle changes, which we can incorporate in our daily routine. This, I would like to acknowledge our university for providing the intramural funding to carry out the few studies which I have mentioned. And also acknowledge my students Adil, Supriya and Bhumika, who have helped me in conducting these studies. Thank you.
Thank you, ma'am. Thank you for your wonderful talk and sharing your research experience. Now the forum is open for discussion. Participants can text your queries on the chat box, which will be addressed appropriately. There's a question from Dr. Usha. Can fasting decrease the risk of cancer? Over to you, ma'am.
Pardon, does fasting, is it?
Can fasting decrease the risk of cancer?
Yeah, there are reports on this, but fasting, it's like intermittent fasting, which has shown, I mean, it has been, I think 2 years back, the Nobel Prize also has been given for this area, where an intermittent fasting has been shown to have a lot of health benefits. So definitely, yes, the fasting would be beneficial for individual because the cells, cancer cells require glucose to grow. So fasting will reduce the supply of glucose to the tumor cells. So definitely the fasting would decrease the nutrient supply to the tumor cells, and the tumor cells will not grow, and there would be, the reduction in the size of the tumor as well, or the normal cells may not progress to tumor cells because of the lack of nutrition here. But again, healthy nutrition is something which we have to keep in mind. So fasting cannot be long. Intermittent fasting is something which has been recommended.
Thank you, ma'am. Ma'am, can you elaborate on the pandemic of COVID-19 as a confounding variable in the study of obesity as a risk factor for cancer?
Yeah, again, this also has been done by us, and we have seen that individuals with increased BMI had more of admissions to the ICUs, especially in our hospital. So, not a multicentric study, it was done only in our hospital. And we have actually compiled some of pathways or some of the factors which actually help make the obese individuals at risk for COVID infection. Not only COVID infections, of course, we had titled our review article as risk of COVID infections, but any kind of infection or this one can be linked to obesity because of various factors. In case of COVID, why we were more interested because it's AC2 receptors, which is a receptor where the COVID virus binds to and gains entry into the cells, are also present on the adipocytes. So that's the reason why this infection was seen more in case of, severe infection was seen more in case of obese individuals when compared to the normal individuals. I mean, this was a small observation what we had in our center. Mainly because of the presence of this AC receptors on the adipocytes.
Thank you, ma'am. Thank you for the insightful talk. Any other questions? Okay. Thank you, ma'am. And now we'll break for tea for about 15 minutes, and we will reconvene for the next talk on the biomarkers in cancer by Dr. Shashidhar K. sir. Thank you.
Thank you.
Thank you, ma'am.
Thank you very much.
I will welcome back all the participants to the CME titled Current Aspects in Cancer Biology, Current Trends in Cancer Biology. Now I request Dr. K. Shashidhar sir to speak about biomarkers in cancer. I feel proud to say a few lines about sir. Dr. K. Shashidhar sir, Professor and HOD Department of Biochemistry, Vice Principal, Sri Devraj Arus Medical College, Kolar. Sir has got 19 years of teaching experience and 19 years of research experience. To his credit, sir has total 19 research projects in hand, including ICMR, institutional. Sir is PG guide, co-guide, supervisor, and co-supervisor for PhD thesis and collaborative research. To his credit, he has got 68 scientific publications. Sir has technical expertise in diagnostic lab accreditation courses. Sir has many research projects in fluorosis, diabetology, and microvascular complications with impact or stress on lipidology, biomarkers. Sir is Vice Principal, Director of Research and Innovation, Editor-in-Chief, College Magazine, Sankirna. In addition, Sir has various other administrative posts. Notified Fluorosis Research and Referral Laboratory as Center of Excellence by Sri Devraj Arus Academy of Higher Education and Research, Kolar. Over to you, sir.
Thank you, Dr. Sonia. A very good morning to one and all. I welcome you all for today's CME. The concept of the CME is the current trends in cancer biology. Biomarkers in cancer is a topic that has been, I will be talking for another 30 to 35 minutes. So myself, Dr. Shashidhar K. as Dr. Soumya has given an introduction about me. The main objectives of today's discussion is general aspects of the biomarkers, the classification of biomarkers, cancer biomarkers, SOP of acceptance of a biomarker, characteristics of an ideal biomarker, the biomarkers, the surrogate versus the clinical endpoints, the challenges in translating the biomarker research into clinical space, and finally, few things I will be touching upon the non-specific biomarkers. The biomarkers initially, if you see into the history and the present and the future status, the biomarkers are the biological markers, which are the broad subcategory of the medical science. Initially in the past, they used to take the pulse, the blood pressure, and every other detail of the patient, starting from anemia to the jaundice, the clinical signs and symptoms. That were the biomarkers that used to be there. Later on, what happened after the advent of the chemistry and various other advancement in the laboratory process, the investigations and other things, now the biomarker has come into the picture, and the futuristic aspects are also very challenging. The objective indications of medical state observed from outside the patient are the ones which they were used to observe before, and they were used to be measured accurately and reproducibly. As I mentioned, the pulse, the blood pressure, the basic chemistry, and the laboratory investigations of the blood and tissues were the one that were used before. And as on date, the normal biological processes, the pathogenic processes, or the responses to an exposure or intervention, including the therapeutic interventions, are in vogue. The future biomarkers shall be able to define the effects of the treatments and interventions which are in the progress. There are various steps that are driven for the biomarker for the targeted cancer therapy to improve the patient outcomes. So first, there should be a request from the clinician that we need to have a biomarker for a certain disease or a cancer or something like that, this will enable a researcher or a clinician to think in terms of developing a reliable biomarker in association with various other fields and branches of the medicine. Once it has been diagnosed with the cancer, a biopsy shall be performed by the pathologist or the clinician. It may be ultrasounded or it may be direct. Then followed by the provider orders appropriate test and the clinical facility, which is equipped with the testing infrastructure. And with the advent of the various advanced research that has been done with respect to the instrumentation and the technology, so it can be taken care as on date. Most of the challenges that have been faced by the clinicians are addressed by the laboratory and the researchers. Further, these investigations should be able to be covered by the insurance and various other things, because the cost of these biomarkers, when it has been thought of a research molecule, will be of very high cost till it becomes a diagnostic molecule. Once it has been done, the test is performed, interpreted, and identifies a therapeutic selection of a biomarker, and finally the patient receives a targeted therapy. The categorization of the biomarkers has been classified into diagnostic markers, the monitoring biomarkers, pharmacodynamic markers, the response to therapy biomarkers, predictive biomarkers, prognostic biomarkers, susceptibility or risk biomarkers, and safety biomarkers. They can be used alone or in combination. Usually, it is a multimodal approach that has been taken to assess health or disease state of an individual. Every biological system, example, the cardiovascular system, the metabolic system, or the immune system, have specific biomarkers in vogue. The diagnostic biomarkers are used to confirm the presence of a disease and the subtypes or a medical condition. The monitoring biomarkers are used to assess the presence status or extent of a disease or medical condition to evaluate the response to an intervention. The pharmacodynamic response biomarkers are applicable to evaluate the response to a medical condition or clinical intervention. The predictive biomarkers are really challenging to identify the probability of develop a clinical event, whether it is a positive or a negative after the exposure to a medical product or an environmental agent. This challenge even we have faced with respect to the highly fluoride rich area here in this area. The prognostic markers are used to identify the likelihood of a clinical event, disease, recurrence or progression in patients diagnosed with a disease or having a medical condition. The susceptibility or the risk biomarkers are used to measure the risk of individual to develop a disease or medical condition in patients without the disease or medical condition. The safety biomarkers are the ones which are used to predict the toxic adverse events induced by the drug, a medical intervention, or environmental agent exposure. Again, with respect to these safety biomarkers, it has been observed in the local population that Kolar, once it was known for the gold fields, where the silica, asbestos, and various other things were exposed to the population, the local population of the KGF, which we call it as Kolar gold fields, which is around 30 kilometers from our institution. Their majority of the population were exposed to these silica, asbestos, and various other things. No doubt, there was chances of lung cancer. In addition to the lung cancer, majority of the population were diagnosed to be having diabetes in this local population. So how come these people, whether the genes are mutated and have become protective, or were they resistant to undergo mutation, were the challenges that we had faced. However, we have observed by conducting various population studies, which was done by the Department of Community Medicine and has got published in various reputed journals, it has been documented and observed that the diabetes is high in this population compared to the lung cancer and various other cancers where the asbestos, silica, and various other things have been exposed with. The US National Academy of Sciences has categorized the biomarkers into three things based on the exposure, response or effect, and susceptibility, which has been followed by the various characteristics, clinical decision making, genetic and molecular biology methods. Let us discuss one after the other, starting from the characteristics. The characteristics biomarkers were those which were used with respect to imaging, molecular, as well as nucleic acid-based biomarkers. These nucleic acid-based biomarkers are the present-day challenges many of the clinicians as well as the researchers are facing with, including the laboratory physicians. The imaging biomarkers include the CT scan, the PET scan, and the MRI. The molecular biomarkers are the non-imaging biomarkers, and they help in measurement in the biological samples, including the CSF, serum, plasma, bronchio-alveolar lavage, as well as biopsy, tissue, and liquid for molecular testing, which this liquid for molecular testing, liquid biopsies, and various other things will be discussed by my fellow colleague, Dr. Anirban Chakravarti, subsequently. The nucleic acid-based biomarkers includes the gene mutations or the polymorphisms, quantitative gene expression, molecules that are included under the characteristics biomarkers. The decision-making biomarkers are used in early drug development. Pharmacodynamic biomarkers of special interest in dose optimization studies. The genetic and molecular biology methods includes 3 types: type O, type 1, and type 2. The type O biomarkers, the genetic and molecular biology markers, includes the natural history markers, where the natural history of a disease and correlates longitudinally with known clinical indices, where already I have given the example of the silica and asbestos that we had experience with. The type 1, the drug activity markers, captures the effect of a therapeutic intervention in accordance with its mechanism of action. The type 2 includes the surrogate markers, which are intended to substitute for a clinical endpoint, however, with limitations of its own. A surrogate endpoint is expected to predict clinical benefit or lack of benefit on the basis of epidemiology, therapeutic, pathophysiological, or other scientific evidence. The predictive biomarkers define the populations that might respond more favorably to a particular intervention from an efficacy or safety perspective. The one example which we have done with respect to the local population I wish to give here is about the NGAL, neutrophil gelatinase associated lipocalin, which is being used in diabetic nephropathy. The studies have predicted that this NGAL is supposed to be a very important and a predominant marker with respect to the chronic renal failure. However, we did it with respect to the acute kidney injury, and we have got a positive predictive value. And this acute kidney injury we identified with respect to the snake bite, organophosphorus poisoning, myocardial infarction recovery patients, and various other conditions which are involved with respect to the acute kidney injury. The diagnostic biomarkers define a population with a specific disease. For example, the cardiac troponin, which is used in the diagnosis of myocardial infarction. The prognostic biomarkers are those cancer biomarkers which are used for monitoring the clinical response to an intervention. Example, HER2 or NEO in breast cancer, EGFR expression in colorectal cancer, HbA1c in assessing the long-term control of diabetes mellitus, which our institution is doing with the help of immunohistochemistry section of the Department of Pathology. The World Health Organization describes the use of multiple biomarkers with a multimodal approach, the prevention and prediction biomarkers, personalization and cooperation biomarkers as the future and challenges in use of biomarkers in clinical medicine. Once there is a request from the clinicians, this will give a food for thought for the researcher to help in discovery, verification, and validation. And this will be done with the help of the various sections of the medical institutions, which includes a pathologist, a pharmacologist, a biomedical engineer, and various other people, the technical people who are involved with respect to the research on the clinical biomarkers. However, whatever it may be, so finally, there will be utilization of the liquid chromatography with mass spectrometry at each step of discovery, verification, and validation. Once this is done, an SOP has to be prepared for acceptance of a biomarker, which shall include the discovery or identification of a biomarker, confirmation, then validation and refinement or established relevance to the population. Here, the major challenge with respect to the validation is the proficiency testing providers with respect to the newer biomarkers, where a lot of other community-related health studies needs to be done, and we need to define our own value with respect to the validation and refinement or established relevance to the population. Finally, it will be adopted, identified, and it will be clinically utilized. Few examples I wish to quote here are the HbA1c, CA125 with respect to the ovarian cancer, HbA1c with respect to long-term diabetes control, which once was used as a marker, and even still it has been used as a marker to assess the mean blood glucose as well as long-term diabetes control of over the lifespan of the RBC. CA125 in ovarian cancer, CKBB, which was used with respect to the prostate cancer and brain tumors, where the specificity is a concern with respect to this molecule. Thus, the PSA has been used for the as a biomarker for the prostate-related diseases as well as prostate cancer. The characteristic features of an ideal biomarker includes it should be appropriate for checking a particular disease condition. It should be safe and easy to measure. It should be user-friendly, cost-efficient to follow up. It should be modifiable with the treatment because we need to assess the prognosis of a disease, how the disease is getting progressed, whether the biomarker values are helping in predicting the improvement or worsening of the condition. It should be consistent across the gender and ethnic groups, because of late we have observed that most of the biomarker studies, the clinical trials and various other things are done either in the Western countries, where the Indian population incorporated as a part of the routine medical examinations. The key issues that have been observed with respect to the biomarkers are the determination relationship between the measurable biomarker and the relevant clinical endpoints. The cancer biomarkers are the substances or a process that is indicative of the presence of a cancer in the body. It may be a molecule which is secreted by a tumor or released in specific response to the presence of the tumor in the body. There are 5 modalities for diagnosis, prognosis, and epidemiology of cancer, which includes the genetics, the epigenetics, the proteomics, the glycomics, as well as the imaging. An ideal biomarker should be able to be assessed in the body fluids, such as it should be available to be estimated in the blood, the serum, the sweat, or various other body fluids, including the stool sample, that urine sample, as well as the stool sample that we collect from the patients. The biomarkers, the cancer biomarkers, can be classified under 3 major headings. Number 1, based on the disease state. Number 2, based on the biomolecules. Number 3, based on other criteria. Based on the disease state, they can be subcategorized into prediction biomarkers, detection biomarkers, the diagnostic biomarkers, as well as the prognostic biomarkers. Based on the biomolecules, the DNA biomarkers, RNA biomarkers, protein biomarkers, and glycome biomarkers. Based on other criteria, it shall include imaging biomarkers, pathological biomarkers, and in silico biomarkers, and this in silico biomarkers will be discussed later by various other speakers. The DNA, as we all know, the genetic material, if there is any mutation in the DNA, that will be translated, that will be transcripted and translated to the RNA as well as to the protein moiety, where the proteomics plays a very important role. The mutations can be observed even in the DNA, RNA, as well as various other molecules, the genetic material that are available. The lipids of the cell membrane and the metabolites, the end product of the metabolism, also plays a critical role.
What happened?
However, whatever the discovery that has happened with respect to the cancer biomarkers, it has been observed that various people who are suffering from the cancer do have various queries with respect to the prognosis, diagnosis, prediction, pharmacodynamics of the drugs that has been given, and the recurrence. For example, the BRCA gene with respect to the breast cancer, or PSA with respect to the prostate cancer. The patient's perspective, a cancer patient's perspective, is likely to know about the development of this cancer, how much prognostic value does this have, the diagnosis, what type of cancer is it and in which stage it is, and is this the optimal drug that is given with respect to the cancer, and will this biomarker will help in prediction of the staging of the cancer and various other things, and what is the optimal dose that is required for the persons who is suffering from the cancer, and will this cancer return or will there be any recurrence. These are the major queries that are asked by the patients and which are posed by the clinicians to the laboratory physicians or to the researchers. So each one needs to be addressed by the clinician with the help of the researcher as well as the laboratory physician. The definitions of the cancer biomarkers are defined by various other people. However, it often is used to cover any molecular, biochemical, physiological, or anatomical property that can be quantified or measured. The National Cancer Institute defines biomarker as a biological molecule which is found in any of the biological fluids or tissues that is a sign of a normal or an abnormal process or a condition or a disease. It should be easily measurable, either invasive or non-invasive, with the help of the various non-invasive biological fluids such as the urine, stool, and various other things. Or, if required, it should be able to be given a predictive value by measuring it in serum or plasma, CSF, or various other biological fluids. A biomarker may be used to assess the prognosis and response to the treatment. Hence, these biomarkers are often also called as the molecular markers and a signature molecule, where it is an authority to say that it is this disease, it is this cancer, and is in which stage it is, and how the person is responding to the therapy. Biomarkers are used in three primary ways in cancer research and medicine. Either it can be used for a diagnosis, prognosis, as well as for the prediction. That is to identify the early stage of cancer, to forecast how aggressive a condition is, and determine the patient's ability to fare in the absence of the treatment. Once the treatment has been withdrawn, how better these biomarkers are helpful in predicting whether the cancer is progressing or has it been arrested, whether the apoptosis has been happened. These are the things which it should be able to predict. The role of biomarkers with respect to the cancer medicine as well as the cancer research are, it should be able to help in diagnosis, risk assessment, screening with respect to the field population, patient stratification, monitoring the therapy, staging, as well as prognosis. So, as I mentioned, it should be able to identify the risk assessment, diagnosis, prognosis, treatment predictions, pharmacodynamics and pharmacokinetics, monitoring treatment response, as well as recurrence. The risk assessment biomarkers are associated with genetic mutations or the epigenetic alterations, which offer a quantitative way to determine when individuals are predisposed to particular types of cancers. Few noted examples I wish to give here are the mutations on the gene KRAS, P53, epidermal growth factor receptor, ERBB2 for colorectal, esophageal, liver, and pancreatic cancers. Mutations of genes BRCA1 and BRCA2 for breast and ovarian cancer, abnormal methylation of tumor suppressor genes P16, CDKN2B, and P14ARF for brain cancer. This methylated site tumor suppressor genes are the present day research that are being concentrated of late. Hypermethylation of MYOD1, CDH1, and CDH13 for cervical cancer, and P16, P14, and RB1 for oral cancers are the research that has been done for the risk assessment. The diagnostic markers determine whether the tumors are of the primary or of the metastatic origin. The researchers can screen the chromosomal alterations which are found on the cells located in the primary tumor site against those found in the secondary site. The secondary tumors can be identified as metastatic, whereas if the alterations differ, the secondary tumor can be identified as a distinct primary tumor. For example, the circulating tumor DNA due to the tumor cells that have gone through the apoptosis and can be detected in biological fluids such as blood, serum, plasma, saliva, urine, as well as the stool sample. The possibility of identifying an effective biomarker for early cancer diagnosis has recently been questioned in light of the high molecular heterogeneity of the tumors which are observed by next generation sequencing studies. The prognosis and treatment predictions can be useful in determining the aggressiveness of an identified cancer as well as its likelihood of responding to a given treatment. Few noted examples I wish to give here are the metallopeptidase inhibitor 1, a marker associated with more aggressive forms of multiple myeloma, where the kinetics of this enzyme will help in assessment of the tumor progression, response to the therapy, and various other issues related to the multiple myeloma. The elevated levels of ERPR, that is the estrogen receptor and progesterone receptor expression markers, are associated with better overall survival in patients with breast cancer. This marker has been studied thoroughly and a lot of research has been done with respect to the local population by the various departments of our institution. HER2 NEU gene amplification, a marker indicating a breast cancer, will likely respond to trastuzumab treatment. Mutation in exon 11 of the proto-oncogene c-KIT, a marker indicating a gastrointestinal stromal tumor, will likely respond to imatinib. Mutations in the tyrosine kinase domain, again, the kinetics of the tyrosine kinase plays a very important role with respect to the EGFR1, a marker indicating a patient's non-small cell lung cancer, which is likely to respond to geftinib or erlotinib treatment. The pharmacodynamics and the pharmacokinetics can also be used to determine the most effective therapeutic regime for a particular cancer, because each person's genetics differs, and the population to population, there is a difference in the genetics, and even in the same family, if you look into the pedigree chart, there are a lot of genetics as well as the epigenetics plays a very important role. These are some of the challenges where in a family of 4, 2 may be suffering from cancer, 2 may not be. Why these things needs to be studied in depth, and the pharmacodynamic or the pharmacokinetic response to the therapy is also a present day challenge. People metabolize or change the chemical structure of the drugs differently. Another challenge that is being faced with respect to the cancer biomarker studies. Decreased metabolism of certain drugs and can create dangerous conditions where certain vital organs are affected, in which a high level of the drugs accumulate in the body is another challenge that needs to be done, where a biomarker plays a very important role in assessing the prediction as well as the kinetics of the enzymes that needs to be studied in depth. Drug dosing decisions in particular cancer therapy can benefit from screening for the cancers. One simple example I wish to give here is about the enzyme thiopurine methyltransferase. Mutations in the TPMT gene, which is unable to metabolize mercaptopurine used in the treatment of leukemia and causes a fatal drop in WBC count in these people. So the kinetics of this drug needs to be studied and it needs to be well understood, including the enzyme thiopurine methyltransferase before administering this drug with respect to the leukemia patients. Otherwise, there will be fatalities will be more with respect to drop in the WBC count in these people without observing or without studying about this molecule. Monitoring treatment response, where these biomarkers have shown utility in monitoring how well a treatment is working over the time and whether the patient is improving or not, whether the condition is becoming worsened, where in which stage the person is, and how the successful biomarker have the potential of providing significant cost reduction in patient care, hospitalization days, and the expenses that the patient is going to bear, and next generation, how it is going to be spread, whether it is going to be spread from parent to the offspring. All these things can be studied with respect to this. The simple example I wish to give here is about the protein biomarker S100 beta, which is used in monitoring the response to the therapy of malignant melanoma. Melanocytes produce high concentrations of S100 beta, a molecule, a protein of interest, which is dependent on number of the cancer cells. The response to treatment is associated with reduced levels of S100 beta, where a negative correlation should happen when a person is being treated for this malignant melanoma. The laboratory research has shown tumor cells which are undergoing apoptosis can release cellular components such as the cytochrome C, nucleosomes, cleaved cytokeratin 18, and E-cadherin. These macromolecules are found in circulation during the cancer therapy by providing a potential source of clinical metrics for monitoring the treatment. With respect to the recurrence, the oncotype DX, which is used for the breast cancer assay, where the oncotype DX looks at a panel of 21 genes in cells taken during the tumor biopsy. They predict the likelihood of the breast cancer recurrence. This test is intended for women with early stage and high risk population, where the stage 1 and stage 2 can be identified, and further with respect to the node negative, ERPR negative, positive, invasive breast cancer treated with hormone therapy. The results are given in the form of a recurrence score indicating the recurrence at a 10 years rate. The uses of the biomarkers in cases in cancer research include developing drug targets as well as surrogate endpoints. With respect to the developing drug targets, these biomarkers are used throughout the cancer drug discovery process. If you go back to the history and look into the 1960s, researchers discovered patients with chronic myelogenous leukemia possessed a particular genetic abnormality on the chromosome 9 as well as 22, dubbed the Philadelphia chromosome. Later on, as the days progressed, the researchers have observed that chromosomes 9 and 22 combine together and they create a cancer-causing gene called as BCR-ABL. This BCR-ABL gene acts as a principal initial point in all of the physiological manifestations of the leukemia. The BCR-ABL gene stratifies a certain subtype of the leukemia, and the drug developers were able to develop imatinib, which effectively inhibited the BCR-ABL protein and significantly decreased the production of cells containing the Philadelphia chromosome. I wish to say a few things about the biomarkers, the surrogate endpoints versus the clinical endpoints. The clinical endpoint biomarkers characterize how a subject in a study or clinical trial feels, functions, or survives. However, with respect to the surrogate endpoint biomarkers, it has been observed that they are the substitute for a clinical endpoint. However, they substituted only when there are solid scientific evidences such as the epidemiological data, therapeutic data, and or the pathophysiological factors that consistently and accurately predicts a clinical outcome, either a benefit or harm. These surrogate endpoint biomarkers serve as a stand-in, but they are not the replacement of a clinical endpoint, because always the laboratory findings should be read under the light of the clinical findings, and they should be supportive to the clinical endpoint rather than taking a ultimate decision with respect to the cancer staging as well as the therapeutic response. The surrogate endpoint biomarker should be capable of being measured objectively and reproducibly, but the two major challenges that are the clinician, that the laboratory physician faces with respect to surrogate endpoint biomarkers are the validation of the values. One is the internal validation as well as the external validation. The limitations of the surrogate biomarkers: these markers present substantial risks when trial designers confuse them with clinical endpoints. They serve as true replacements for clinical relevant endpoints. They have as ultimate measures clinical outcomes, example, retrospective analysis of a biomarker. They require continual re-evaluation of the causal relationships between the surrogate endpoints and the true clinical endpoints, and they need constant validation, both internal as well as external. These are some of the limitations with respect to the surrogate biomarkers, whereas the clinical biomarkers, clinical endpoint biomarkers, these things are not there, where the clinical findings itself will help as a biomarker. Say, for example, the pulse rate, the anemia, the jaundice, or the yellowish discoloration of the sclera and conjunctiva, various other things will help. The cyanosis that is observed, these things will help in diagnosis of the underlying cancer with respect to an individual. The cancer surrogate endpoints application: biomarkers acts as a stand-ins for the effects of a drug on cancer progression and survival. Use of validated biomarkers prevent patients to undergo tumor biopsies and lengthy clinical trials to determine if a new drug is worked. In the current standard of care, the metric for determining a drug's effectiveness is to check if it has decreased cancer progression in humans, and untimely, whether it prolongs the survival. The successful biomarker or the success of a surrogate biomarker could save substantial time, effort, and money if failing drugs could be eliminated from the development pipeline before being brought to the clinical trials. This is one of the major challenges where majority of the pharmaceutical industries are facing as on date. Ideal characteristics of surrogate endpoint cancer biomarkers should be involved in process that causes the cancer. The changes in biomarker should correlate with changes in the disease. Levels of biomarker should be high enough that they can be measured easily and reliably. Levels or presence of a biomarker should readily distinguish between normal, cancerous, and precancerous tissue. Effective treatment of a cancer should change the level of the biomarker. Level of the biomarker should not change spontaneously or in response to other factors not related to the successful treatment of a cancer. So, the ideal characteristics of the surrogate endpoint cancer biomarkers plays a very important role with respect to the cancer research. The two areas of interest as surrogate biomarkers include the circulating tumor cells as well as the circulating miRNAs. Both these markers are associated with the number of tumor cells present in the blood, and as such are hoped to provide a surrogate for tumor progression and metastasis. Significant barriers to their adoption include the difficulty of enriching, identifying, and measuring CTC, that is the circulating tumor cells, and microRNA levels in the blood. New technologies and research are likely necessary for the translation into clinical care. The types of cancer biomarkers includes the molecular cancer biomarkers, and certain examples I wish to give you are the notable examples includes the ERPR, there's the estrogen receptor and progesterone receptors biomarkers, and HER2 NEO receptors with respect to the breast cancer, where some of the biomarkers are used for the diagnosis of more than one organ or one cancer. EGFR, KRAS, UGT1A1 is used as a biomarker for the colorectal cancers. HER, NEO is used for the gastric cancer. JAST can be diagnosed by using the CKIT. CD20, CD30, Philadelphia chromosome, as I mentioned, 9 and 22, that is the mutated form of this, that is the BCR-ABL, and various other markers that has been shown here is used for leukemia as well as lymphoma. EGFR, EML4, ALK, KRS is used in the diagnosis of lung cancers. BRF, with respect to the melanoma, elevated levels of the amino acids, leucine, isoleucine, and valine is used with respect to the pancreatic cancers. BRCA1, BRCA2, with respect to the tumor suppressors lost in the cancer. mRNA, microRNA with respect to the RNA-related tumors, prostate-specific antigen, which are found in the body fluids or the tissues used in the diagnosis of prostate cancer, and CA125 with respect to the ovarian as well as the breast cancer. Merkel cell polyoma virus or the antibodies which are used as antigens, cDNA with respect to the DNA. There are certain markers, cancer biomarkers, having less specificity or without specificity, because these, not all cancer biomarkers have to be specific to the types of cancers. As I have told, they may be coded for 2 or more than one cancer, as these are some of the limitations of these biomarkers. These biomarkers are found in circulatory system, can be used to determine an abnormal growth of cells present in the body. Biomarkers for cancers can be identified through diagnostic blood tests, which is one of the main reasons to get regularly health checkup of the high-risk population or the pedigree chart where the family history of cancers are there. The non-specific biomarkers includes the NLR ratio, BFGF, and IGFR, which has been spoken in detail about this insulin-like growth factor by Dr. Akila Prashant. The NLR, neutrophil to lymphocyte ratio, helps in the prediction of the cancer. The basic fibroblast growth factor, insulin-like growth factor, has its own applications as well as its limitations. The neutrophil to lymphocyte ratio, that is the NLR ratio, is a non-specific determinant for the cancers. It mainly focuses on the activity of two components of the immune system, that are involved in inflammatory response, which is shown to be higher in the presence of malignant tumors. The basic fibroblast growth factor, that's the BFGF, is a protein that is involved in the proliferation of the cells. This molecule has been, or this protein has been shown that in the presence of the tumors, it is highly active, which has led to the conclusion that it may help malignant cells produce faster, reproduce at faster rates. The antibodies to this anti-BFGF can be used to help treat tumors from many origins. The insulin-like growth factor is involved in cell proliferation and growth. It acts as a growth factor. IGFR is involved in inhibiting apoptosis, which is nothing but the programmed cell death. The levels of IGFR can be increased when cancer, such as the breast, prostate, lung, and colorectal cancers are present. To say a few things about the overview of these non-specific biomarkers, NLR, there are no biosensors available for it as on date, and it can be estimated by the routine investigations, either by using a 5-part analyzer or by doing the manual, counting the manual cells of these things. These are elevated with respect to in reflex response to the inflammation caused by the cancer. Basic fibroblast growth factor has got an electrochemical method. This level increases when a tumor is present and helps with the fast reproduction of the tumor. The electrochemical impedance spectroscopy sensor helps in the identification or estimation or quantification of or qualification of this insulin-like growth factor, IGFR, which is highly active in cancer cells and which helps in reproduction. The impact of cancer with respect to the local population as well as to the as a global issue, it affects the society worldwide, and by testing these biomarkers, early diagnosis can be given to prevent the death. In conclusion, I wish to say two statements here. One is the biomarkers are of great use, which can be used as a diagnostic, predictive, prognostic, and therapeutic, and it can be used in the development of the drug, as well as it can be used as a marker to assess the prognosis of the cancer. The newer biomarkers must be a representative of the clinical endpoints, which has to be validated and re-evaluated constantly for its use in the clinical practice. Finally, I wish to give the take-home message that there is a can in the cancer where we can beat it, we can fight for it. There are markers which can help in early diagnosis, early prediction, know the staging of the cancer, how the person is responding. All these things can be done. Thus, there is a can in the cancer. Let's not lose the hope, and we can, and we all shall fight for it. Thank you.
Thank you, sir, for the informative talk on biomarkers in cancer. Now the forum is open for discussion. I request participants to post doubts in the chat box, which will be addressed. Sir, there is a doubt from Dr. Usha ma'am. Any biomarker specific for epigenetics associated cancer? Any biomarker specific for epigenetics associated cancer?
Yeah. Ma'am, 1 minute. I will just open my chat box. Usha Bear, right? Can fasting decrease the risk of cancer? No, not that one. Any biomarker specific for epigenetics associated cancer? Yeah, depending on the cancer, the epigenetics plays a very important role. There are no specific biomarkers as on date, to my knowledge. However, we can find out with the recent advances that is happening every day, we need to look into much more with respect to the epigenetics of any of the cancer, the causation, the staging, and various other things.
Thank you, sir.
Thank you, Dr. Soumya.
Thank you, sir. Now I request Dr. Chimraj, sir, to present on tumor markers, a diagnostic tool, because every test is a life. To say a few lines about sir, Dr. K. Chimraj, sir, pursued PhD Biochemistry from Mahatma Gandhi Institute of Medical Sciences, Sevagram under Nagpur University. Sir is working as OrthoCare National Lab Support Manager, Ortho Clinical Diagnostics. Sir has got experience of about 29 years in the field of diagnostics industry. Sir has worked on immunodiagnostic of filariasis and tuberculosis infection in the level of research officer. Sir worked as research and development manager, QC manager, and production manager at Dr. Reddy's Laboratories Limited, Diagnostics Division, Hyderabad, and has got 10 plus years of experience. Sir has 18 plus years of experience in orthoclinical diagnostics in providing application support. Sir, to his credit, sir has 18 original research papers in both national and international journals related to immunodiagnostics of filariasis and tuberculosis infection. Sir is honored with several awards like Ortho President Club Award, Ortho Top Gun Award, Ortho Gold Achiever Award, etc. And has got a newsletter, Results Matter, weekly blog in the field of transfusion medicine. Welcome you, sir. Over to you, sir.
Thank you, Dr. Soumya, for the excellent introduction. Good afternoon to all. First of all, we would like to thank Dr. Sashidhar sir and the organizing committee members for giving us an opportunity to present in this scientific session on current trends in cancer biology. So after the excellent presentation from Dr. Sashidhar on this, the biomarkers, so I would like to give you a few practical applicability of this tumor markers. So my topic, it will be the tumor markers is as a diagnostic tool, because every test is a life. Because in this era of evidence-based medicine, laboratory plays a major role in providing evidence for the clinicians, for the oncologist, for the diagnosis, treatment, as well as for continuous monitoring of this disease for its recovery. So we, from orthoclinical diagnostics, our mission is to partnership with our clinical medicine practitioners to improve and save lives with the diagnostics. Because every test is a life. So orthoclinical diagnostics, it's a pioneer in the diagnostic field way back from the year 1939, more than 81 years plus experience in the diagnostic field. And it is a global organization, presence in more than 140 countries with more than 250 products. So if you see that what matters most to the clinicians from the lab, the first expectation is the right results. The results should be accurate, reliable, reproducible, and it should be benefit for them in their clinical diagnostics. And second important aspect is providing the results in the right time. In this fast world, everyone wants the results as early as possible. The so-called turnaround time may be ranging from 1 hour, 2 hour, 3 hour, 4 hour. Within that time they want it. And all it should be within the affordable cost. In the recently, if you see that, there is a change in the role of laboratory medicine. Earlier, it is a specimen-centered clinical testings. When it was only in the Department of Clinical Laboratory. Now, when it is moved to the Department of Laboratory Medicine, the role is changed from specimen-centered clinical testing to the patient-centered clinical decision-making. How the report is helping the clinicians in decision-making that. In the earlier, we used more focus on the clinical laboratory performance. How is my internal quality control performance? How is my external quality control performance? Now the trend is changed from that. Yes, it is important. You have to focus on the IQC, EQUAS, everything that. But ultimately, it's the patient outcomes. So need to focus more on the patient outcomes, how the laboratory reports are helping for the patients. So yeah, it is not only generating the report, it also partnership in the patient care, in continuous monitoring. So this gives, there's no chance of error in this process that. So especially in case of oncology, how the laboratory medicine is supporting the oncologist for making the diagnosis, for the effective treatment, monitoring. So the diagnostic reports generated by the laboratory, maybe using the various tumor markers, it should help for the screening, first of all. It should help in the risk stratification. It assists in the diagnosis. And during the course of treatment, what type of treatment, what is the dosage required? So that the continuous monitoring can happen that, to prevent any recurrence. So that's a role of tumor markers as a diagnostic tool. And we have seen an elaborative presentation from Dr. Sashidhar on these tumor markers. It can be counting the blood cells like a CBC to identify any blood-related cancers. It can be a serum protein measurement like electrophoresis in case of multiple myeloma. It can be the analysis of various biomarkers which can be released by these cancer cells. The various biomarkers like total PSA, free PSA, CEA, CA, 125, 15.3, 19.9, alpha fetoprotein, beta HCG. So there are various markers are there. So how we can able to analyze that and give more reliable and reproducible results. And also there are now the molecular assays available to look for the cancer cell specific genetic materials. So this slide gives the various tumor markers for various type of cancers, which we are all aware about that. So what is its role? As we already seen, it should be used either for screening, monitoring, assist in diagnosis, prognosis, and if there is any recurrence, it should be able to identify it as early as possible. That. So this slide, it gives the various tumor forms and what are the tumor markers which are available, and what is its role, whether it is helpful in the diagnosis, because there is no any ideal tumor marker which can be used. Yes, it is the things based on that we can go for the diagnosis. So still it is our work is going on to identify the ideal tumor marker. But whatever the currently available, can we utilize it effectively? So to have that ideal tumor marker, as well as the ideal tumor marker assay, the basic requirements are it should be highly specific. It should not give the so-called false positive reaction. And also it should be highly sensitive. So that means there should not be any false negative. When there is a tumor is there, yes, is it able to predict or not? So it should have that half-life, the short half-life, so that we can able to see changes in the monitoring during the course of treatment. So it should have the long lead time, so that there is a sufficient time for alteration of the course of disease. And it should be a quantitative type. The level should correlate with the tumor burden, so that it can be used as a marker for prognostic, as well as the predictive utility of the tumor markers. And it should be easy to perform within the short turnaround time. And also it should have the cost containment. And it should be able to do it in the easily obtainable specimen, the invasive or non-invasive. It should be like a serum or other body fluids, so that it is acceptable for the population, the patient population. So from orthoclinical diagnostics, with our vitros analyzer and vitros assays, we have the wide range of assays which can be used as a tumor marker, which can help for the laboratory medicine practitioners to analyze the serum or plasma or blood specimens, to provide the timely report to the oncologist. So in vitros, we have the vitro CEA, CA 99, 15.3, CA 125, total PSA, free PSA, alpha fetoprotein, beta HCG, procalcitonin, beta 2 microglobulin, ferritin, including the anti-HTLV antibody assay, which can be performed in a wide range of systems. We have the vitros chemistry systems, vitros immunoassay systems, and the vitros integration system, where you can able to perform both the clinical chemistry assays as well as the immunoassays. Because various biomarkers can be performed either by means of biochemical methodology or by means of immunodiagnostic methods. And also vitros provide the complete automation solutions, which takes care of all the pre-analytical, analytical, as well as the post-analytical part of the laboratory process, which is called as the vitros automation solution. When compared to the tumor markers, so tumor markers, for the interpretation from this marker assays, you should be able to see the dynamic of such changes. So whether the tumor is developing, even in the early stage, whether there is any increase within the reference range. Initially, it may not be able to detect by CT or PET. It can give some indication, yes, there is a rise in the tumor markers is observed. And that rise is in 3 consecutive blood collections or increased by more than 25% is considered to be a significant changes. And because of its excellent sensitivity to pick up the very low level of these markers with a highly efficient immunoassays, like enhanced chemiluminescence immunoassays. So we can able to detect the tumor of maybe a small, having about 10 to the power of 6 malignant cells. Whereas the symptoms or the clinical diagnosis may be possible when it is increased to 10 to the power of 9 malignant cells. And tumor markers also helpful in the systematic examination when you are doing the monitoring, even post-therapeutic, what is the status of the tumors, as well as during the course of treatment for follow-up. But always when there are assays there is also certain limitations which can impact the accuracy of the report. Since these are all laboratory investigations, there can be certain analytical interferences. The analytical interferences may be related to the specificity of the assay, showing cross-reactivity with the structurally similar molecules. Or maybe because of the hook effect, because sometimes the tumor markers, the concentration can go high. So because of the limitation of the assay, so it may lead to the falsely low values because of this hook effect. Sometimes it can be because of the limitations in the process. There can be a carryover related issues from one sample to another sample, or sample to reagent, or reagent to samples. That type of carryover can occur, which can affect the quality of the final results. And there can be certain pre-analytical errors, the quality of the sample used for screening. The sample may be hemolyzed, having turbidity or icteric, which may non-specifically bind, which can give the falsely high value. Or some of the people may have the interfering antibodies like heterophilic antibodies. And especially in case of cancer patients, when they are under the therapy with some monoclonal antibodies, they may develop this HAMA antibodies, human anti-mouse monoclonal antibodies, anti-mouse antibodies. So these antibodies may interfere, as most of the immunoassays use the monoclonal antibodies for detection of these tumor markers. So there can be an interference from the HAMA, which is the human anti-mouse antibodies. And there can be an human-related errors in the analytical process. So it's always to double check to overcome those errors related that, or the system should be dynamic to identify those errors and predict those errors and prevent those errors. So here in vitros, we use the microwell technology for most of the tumor markers using the immunoassay called the enhanced chemiluminescence assay, because there are various immunoassays are currently in use for the tumor markers. So there are chromatographic card methods are available, ELISA based assays are available, fluorescence based immunoassays are available, chemiluminescence based immunoassays available. So we use that enhanced chemiluminescence immunoassay, which is having the high sensitivity, which can able to pick up 10 to the power of minus 19 moles molecules in the sample. That means very low level of analyte can be able to pick up with more accuracy. And since it is using that luminescence technology where there is a generation of light by means of chemical reaction, so there is no background reaction can be minimized to give more reliable and reproducible test results. So especially when we are having our tumor markers, what's our general expectations? Yes, it should not pick up any, it should be more sensitive, it should be more specific. So there should not be any variation or difference. There should be a clear-cut difference between the healthy population than the patient population who are having these tumors. That's the general expectation. Yes, I'm doing this. When it is coming negative, it should be a true negative. Whatever it is coming as a value above the reference range, then it should be having the tumors. But is it possible? In reality, it may not be a possible that. There is a overlapping. There is a gray area. So none of the tumor markers are having, either you can say that it is 100% sensitive or 100% specific. There is some amount of false reactivity or false positivity or false negativity, which can affect the sensitivity. Usually we calculated the sensitivity. The manufacturers also give that what is the sensitivity of their assay and the specificity of the assay. In other words, we can use it as based on the negative predictive value. So it is how much negative predictive value. When I say that yes, it is negative, it should be above 90%. And the positive predictive value. These are all little statistical calculations for any assays. Especially it is very important in case of the tumor marker assays. So if you see that the Vitro's CE assay, carcinoembryonic antigen assay, which is measured by means of enhanced chemiluminescence immunoassay based on the immunometric assay, where the results will be available within 38 minutes with a sample volume of 20 microliter serum or plasma. So which uses the sandwich type assay where the sample CEA is sandwiched between the biotilated mouse monoclonal anti-CEA antibody, which are already captured in the streptavidin coated wells. On the other side, we are using the horseradish peroxidase labeled mouse monoclonal anti-CEA antibody. So if the patient is having certain HAMA antibodies, so that may not specifically bind to this monoclonal anti-CEA antibody, which can again affect the results. So the assay should be able to prevent those type of interferences. The assay is designed like that. If you see that the results, so especially we have to see that what is the results in the normal population, healthy populations, as well as either the malignant or non-malignant condition that. So here in this, when they have done that validation of this CE assay, so it was taken the smokers and non-smokers, the malignant disease with the colorectal, breast, pulmonary, ovarian or gastrointestinal cancers, as well as the non-malignant disease. So if you say here that almost 91.9% of the healthy non-smokers, they are all having CA with this less than the cutoff value of 3 nanogram per ml. In case of smokers, yes, there is about only 67.3% are having this less than the cutoff point. In case of malignant cases, Yes, almost 67, more than 67% of the people are having this, especially in case of breast cancer. CEA is found to be more than 55%. In case of pulmonary, it is found to be more than 50%. So it's not like a 100% you can able to predict that. That's a message you have to understand that. So CEA is, it's not a cancer specific, but it is cancer associated. Similarly, the level of CEA, when it was found in the different cancer patients, the breast cancer or colon or liver, rectal or stomach. So the mean concentration is high in case of colon and liver cancer with more than 90 units. Whereas in case of other cancers like a breast, which is very low, rectal or stomach cancer also, the nominal increase in the value. Similarly, CA 125, which is also you can able to do by means of immunometric assay, same like a CA, the sandwich assay. The results will be available in 37 minutes. And here also, if you see that in the cancer patients, as well as in the healthy normals, so 98.5% of the population is having the CA 125 below the cutoff level, which is 35 units per ml. Whereas in case of ovarian carcinoma, almost 72% had concentrations above 35 unit per ml. That. So again, here also, it's not a specific to particular tumor, it's a associated. And PSA also, it can be used for the prostate cancer. So we have both the vitros total PSA as well as the free PSA, two different assays, same immunometric assay with the results within 24 minutes. So especially if you see that in case of normal, so majority of them, almost close to 80% are less than the cutoff point. Whereas in case of malignant conditions, so quite a number of cases are having the above, especially here when the values are just above the cutoff point. That means 4 to 10 nanogram per ml, which is the gray area. So in such a cases, alone total PSA may not be helpful. It's better to go for the total and free PSA and see the percentage of the free PSA. So in this study, so about 583 had normal DRE, not suspecting the prostate cancers, but there was certain clinical conditions. So out of this, 283 were having the total PSA in the gray area between 4 to 10. And all those cases were subjected to the free PSA, which helps in better prediction. So when the percentage is low, less than 10%, the probability of the prostate cancer is increased. Because in case of prostate cancer, free PSA level will be very low compared to the total PSA. Whereas in other conditions, maybe free PSA also, it will be on the higher side that time. So the percentage of free PSA, if it is lower, So it can be a prostate cancer. If it is higher, then it can be a benign, not a malignant. So thereby we can able to predict with the help of total PSA and the free PSA with the ratio. Same case in case of CA 99 also. So in the general population, out of 200, 97% are having value below the cutoff. So your negative predictive value will be improved. Out of 50 patients of pancreatic cancer, so 35 samples, so almost 70% are having the value above the cutoff level. Same in case of vitro CA 15.3. So almost in the healthy populations, 98% are having the value less than the cutoff limit of 35 units per ml. And about 94 patient population with the breast cancer. So about 38 had value above the cutoff level that. So approximately close to 50% you can able to predict that. And also it helps in the staging. Also this alpha fetoprotein. So the cutoff is 7.22 because majority, more than 95% of the normal population are having the values less than 7.22 IU per ml. Whereas in case of hepatocellular carcinoma, almost 90% are having the value above the cutoff level that. Especially this alpha fetoprotein, it can be used as a sword with 2 blades. So as an oncofetal antigen, it can be used as a diagnostic marker for liver cancer because this alpha fetoprotein, it promotes tumor proliferation, invasion, angiogenesis, and also escape from the immune surveillance, anti-tumor apoptosis. On the other side, alpha fetoprotein can be used for a vaccination. The alpha fetoprotein modified vaccines like a DNA vaccine or protein vaccines or peptide vaccines or disease cell vaccine, which can be used for the anti-tumor immunity against this alpha fetoprotein positive tumors. So it can have the double value. And the other tumor markers like beta-2 microglobulin, it can be useful in case of cancers related to the bone marrow or blood, which can help to identify the severity of the cancers, especially in the cancer staging. When the alpha beta-2 microglobulin is higher, in the higher stage, so it is in the advanced stage. So it can help to predict the disease development, helping in the treatment, course of treatment, and ensure that whether the treatment is effective or not. Is there any metastasis happening or not? So this case of multiple myeloma, this beta-2 microglobulin, along with the other like albumin, LDH, and the genetic gene abnormalities, so it can help in the staging. So whether it is in the stage 1 or stage 2 or stage 3, based on the revised international staging system, based on the level of beta-2 microglobulin, albumin, and LDH, along with the cytogenetics. So another one is adult T-cell leukemia or the lymphoma. One of the etiologic factor for this adult T-cell leukemia, it can be because of this viral infection, especially this HTLV. So if you see that there are multiple publications from Kerala about this adult T-cell leukemia, lymphoma in the Kerala, and some of the things are found to be confirmed as positive for the anti-HTLV antibody. So which gives the things, is it required to screening all these patients who are having this adult T-cell leukemia? Because usually the prevalence of HTLV infection is, mostly it is not there in India. That's the thing. But there are sporadic publications are there. There is a prevalence of some amount of HTLV is there in the population. So we have the Vitros anti-HTLV 1 and 2 antibody assay on the same platform based on the enhanced chemiluminescence technique, where within 48 minutes we can able to see the presence of this anti-HTLV antibody. So which is having the excellent sensitivity as well as the specificity. So this can be, we have, but it is not much in use in India. Maybe in case of adult T-cell leukemia, this can be also used as a marker to identify the root cause of this leukemia. Okay, these are the different markers. Another important basic difference, this one is how we can able to minimize these laboratory testing errors. Because whenever there is a human intervention is there, there is a errors are there. So even though you are using the high excellent technology, there is a possibility of errors, maybe in the pre-analytical, maybe in the analytical stage. So it was identified that almost about 60 to 70% of the errors are in the pre-analytical and about 19 to 47% are in the post-analytical, because majority of the time the analytical errors are minimized with the quality improvement systems, accreditation systems, follow using the internal quality control and the external quality control performance. So the analytical errors are minimized, but there is no any much control over the pre-analytical part as well as the post-analytical part. Because these things can occur outside the laboratory, starting from the sample collection to the until it is reaching to the laboratory or the processing the samples before loading into the analyzers. So if you see that there is a study which shows the incidence of turbidity, hemolysis and icterus, because sometimes the sample is collected, but sample is having its own anomaly like a endogenous or the exogenous interferences. One of the common interferences is hemolyzed samples, where the RBC host may be there. The turbidity samples, which may not be easy to centrifuge it and clarify that. Especially in the automation systems, when it is used, it may be picking up those particulate matters also, which may not specifically bind, and then it may give the false results. So in this study, they have observed that almost 9.7% of the samples are having this anomaly related to the hemolysis or turbidity or the icteruses. Are we having the capability to minimize these errors? Because these can lead to the errors, since it is a high sensitive assays we are using for the tumor markers. Do we have the control over this? Because visual inspection may not be always possible. And when you are having more than 500 samples, and when it comes with the labels covered, you may not be able to see that, whether it is creating the errors or not. So the CLSI, there is a guidelines, 56A guidelines, which is mainly focusing on this hemolysis, ictericity and lipemic. Because this has to be considered as a quality indicators, because they can able to affect the quality of the results. Are we able to identify which samples are having these interferences? So in the vitro systems, there is a separate systems along with the things without any additional cost. We can able to analyze this hemolysis, ictericity and turbidity of all the samples without any extra cost, without any extra turnaround time, affecting the turnaround time. So it uses that same tip which is used for sample dispensing. The tip is sealed down, used as a cuvette tip, and with that samples, it will be scanning at the multiple wavelength and see the hectare content, hemolysis or the turbidity, and then it posts that the value for each sample. So what is the level of hemolysis? What is the level of ictericity, and what is the level of turbidity? And every assay is having the threshold limit. When it is crossing the threshold limit, then it will also flag the results. So thereby we can able to, first thing is identify which are the samples are having the anomalies and whether is it interfering in my assay or not. So that type of control over the pre-analytical errors is possible with this vitros, with the help of this microsensor technology. And second important error which can occur is because of this carryover in this analyzers, which are fully automated analyzers. Because the probe, it can be some of the things, can be a fixed probe which will go for the washing and then go to the next sample. That the reagents may probe, it can be taken the reagents and then get washing and then it go to the next reagents. So there can be a sample to sample carryover. One high value samples and the next sample may be a normal, but because of the high value in the first sample, some small amount of remnants of molecules can be get transferred to the next sample, that which may lead to the falsely positive results. That especially it is very important in case of this tumor marker assays, because the washing may not be having the efficiency. Not only that, it can also affect the lead to the dilution, because there will be a small volume of the samples, 20 microliter. So if the wash buffer is added in that, which is having almost even 2 microliter in 20 milliliter, which is 10% variation results can be there. So sample to sample carryover is one of the important error. Sometimes it can also happen in the reagent to reagent. So reagents may get contaminated and affect the final results. There is a possibility of sample to reagent. Again, reagents get contamination, or reagent to the samples carryover. But in the vitro systems, we use the disposable tip called vitros Bessa tip and the vitros micro tip. All are disposable. So there is no any carryover related issues, because every sample will get the fresh tips, every reagents will get the fresh tips. So carryover related issues can be minimized or eliminated. That in addition to that, it is having the another technology called as the IntelliCheck technology, which is nothing but to minimize those pre-analytical and the post-analytical errors. It's like an internal auditor. So every process in the automation systems is monitored, including the temperature. In case of vitro systems, because all these immunoassays, you need the washing steps before adding the conjugate, or removing the conjugate before adding the substrate. So even that wash buffer is also preheated to 37 degrees centigrade to prevent any thermal shock to the antigen antibody reaction. That so that's taken care by this IntelliCheck technology, which monitors every process starting from the sample, whether the sufficient sample is having any clot or bubbles. So until the results is delivered, including all the reagents verifications. So every step is verified, and it gives the report called as the Intelli report, which have the traceability, which includes the what is the reagent lot, what time it was performed. So what type of errors, anything is there, it will able to, we can able to predict that. So it's a real-time quality verification for every process. That so this microsensor technology with the disposable tip metering technology, as well as with the IntelliCheck technology, the laboratory errors will be minimized, both in the pre-analytical and the post-analytical part, which helps in improving the accuracy of the report, which is required by the oncologist for all these cancer markers. So in conclusion, the vitros assays, the various tumor marker assays. With the vitros system, based on the enhanced chemiluminescence technology, along with the microsensor technology, IntelliCheck technology, and disposable sample metering systems, with the world-class AutoCAD surveys, you can able to give all the laboratory medicine practitioners can able to give the most reliable, reproducible results with a short turnaround time to the clinicians, especially the oncologists, for the effective diagnosis, screening, monitoring, as well as to identify whether there is any recurrence is happening or not. So with that, I conclude my session. So ortho is the ideal partner in case of cancer, tumor markers for the laboratory medicine. So thank you very much for giving me the opportunity. Thank you. So if there is any questions, I'll ready to take it.
Thank you, sir, for the informative talk. The forum is open for discussion. I request participants to post queries on the chat box, which will be addressed.
Thank you.
Thank you. Thank you, sir. Now I request Dr. Anirban Chakraborty, sir, to present on liquid biopsy in cancer management. To say a few lines about sir, Dr. Anirban Chakraborty is a Director, Dean Faculty of Biological Sciences, Nitte University Center for Science Education and Research, Mangalore. Sir has been honored with Best Researcher of Nitte, deemed to be university in the year 2018, and Best Researcher of Nitte University Center for Science Education and Research, 2018 and 2020. Sir has been awarded with Early Career Research Award by Science and Engineering Board, Department of Science and Technology, Government of India in the year 2016. Also awarded with postdoctoral fellowship for foreign researchers by the Japanese Society for Promotion of Science between 2006 and 2008. Sir's research interests in lab works on dynamics and disorders of ribosome biogenesis using the zebrafish model system. Focus is on identification of non-canonical roles of ribosomal proteins and ribosome biogenesis associated proteins in disease development, particularly cancer. And recently, his team has successfully developed a zebrafish mutant for PARN using CRISPR-Cas9 as a part of DST SERB project. His lab is also interested in rare genetic disorders and has carried out several projects on craniofacial dysostosis. Sir's another area of research is in his group involves developing non-invasive and alternative DNA-based molecular tools for detection of specific variations in Scientific Reports 2020, Journal of Carcinogenesis 2020, etc. Sir has published 65 research articles and has 3 published patents with a cumulative impact factor 176.57 and has got 9 total external research grants handled as a principal investigator and co-principal investigator, among which 6 are completed and 3 projects are ongoing. With this brief introduction, I welcome you, sir. Over to you, sir.
Thank you. Am I audible? Hello? Hello? Am I audible? Hello? Am I audible? Can anyone please confirm?
Yes, yes, please. Please go ahead.
Yes, sir.
Yeah. So I will share my slide. Yeah. Is my slide visible?
Yes, sir.
Okay. Should I start? How is it? Hello?
Please go ahead, sir.
Yeah. There's a bit of okay. Can someone confirm if my slide is moving?
It's visible, sir.
Is it moving as well?
Please give me a slide show, sir.
Yeah, that's what I'm doing. Is it moving? Are the slides moving? Can anyone confirm that?
No, sir. No, sir, not moving.
Okay.
It's visible now, sir.
Visible, but the slides are not moving. That's what you're saying.
Yes, sir.
Okay. Okay. Are the slides moving now?
No, we are not seeing any slides.
They're not seeing any slides. Okay. Are you seeing any slides now?
No, sir. Ah, now, yes.
Okay. And is the slide? Oops. I don't know why the slide is moving. Okay. Okay, it should be able to move now. Is it moving now?
No.
No. Okay. Well, hold on. Okay. I think there's an issue with the slideshow, so I'll just go with the navigator bar. Okay. Let me start by thanking the organizers, Dr. Shashidhar and Dr. Harish, for giving me the opportunity. So let me quickly introduce my topic, which is liquid biopsy. 12:50 lunchtime for most of the people. I don't know how much attention we'll have for this, but anyway, I'll try to keep it interesting and engaging. So this is my topic, which I'm going to introduce you to the audience here. It is called liquid biopsy, which is a very recent and it's a kind of a emerging technique in cancer diagnosis, prognosis, and management. So I think most of the esteemed speakers have already spoken about the cancer as a disease. I don't want to get into the introduction part. Let me come straight to why liquid biopsy is necessary in today's context. And for that, let's see what are the challenges that are with traditional biopsies. So of course, we all know that a cancer diagnosis, I think the gold standard is a histopathological evaluation of the tumor tissue. But for a traditional biopsy to happen, there are many difficulties. One is, of course, the sample has to be obtained by surgical intervention, something if it is growing inside in any of the internal organs. And for that, it is expensive and it is invasive. And the tumor has to grow to a certain size before it is actually considered to be a suspected malignancy. And it is any surgery or any surgical intervention is hard on the patients, both emotionally, physically, and financially as well. And it is not always good for effective long-term monitoring, particularly for lung cancer, because repeated biopsy is a big issue, a big impediment in lung cancer. So there is a need for rapid, cost-effective, sensitive, and of course, the most important part is a non-invasive method, which is necessary for biopsy. So this is where liquid biopsy comes into play. Liquid biopsy basically is defined as the detection of cancer from blood or any other body fluid. It could be saliva, it could be cervical fluid, it could be even sweat or any other body fluid. But generally, most of the people associate it with detection of cancer from blood. It's a non-invasive technique, or you could say it's minimally invasive because you still need to draw a few ml of blood from the peripheral brain. The most important part in liquid biopsy is that it relies on tumor-derived components which are in circulation, and they are called as circulating nucleic acids. So it could be nucleic acids, it could be DNA, or it could be RNA. So these nucleic acids are basically from all tumor-derived components that are present in blood. In addition to nucleic acids, we know that there are exosomes which are present. A lot of research is happening on that. We also have tumor-educated platelets which are released into the bloodstream. And of course, we have circulating tumor cells, which are, which was the initial stage where people started liquid biopsy based on that. And from tumor cells, they have moved into nucleic acids. Now, the liquid biopsy is quite happening in today's context. And if one asks what are the questions one could answer or what could get from liquid biopsy, so it helps in understanding if the cancer is present, first of all, and if it is present, where it is. So how does it tell us from a peripheral blood? Depending on the type of signature mutation that we check, now, depending on the kind of gene that is mutated, depending on the kind of tumor or the mutation that we are seeing, it could give an idea on whether it is a breast cancer or a lung cancer or a colon cancer and so on. It also gives us an idea whether the cancer is spread to certain organisms. It gives us what are the genetic changes that the tumor have, because it relies on products that are obtained from the tumor. It also tells us what are the treatments that could work, particularly important in prognosis, whether treatment is working in the patient or it is not happening. And finally, it also tells us if there is any cancer left after treatment. Generally, PET scan is usually done to determine the cancer-free regression or cancer-free survival stages. But recently, liquid biopsy has also been shown to predict to a certain extent whether the cancer has or the patient has gone into remission. So liquid biopsy of cancer using cell-free DNA, we call this cell-free DNA, which are basically DNA which has released into the bloodstream. These are fragmented pieces of DNA which are not associated with any cell. These are free DNA which are present in the, generally we see them in plasma or the serum of the blood. And it is also known that patients with cancer usually have higher levels of these plasma or serum level compared to patients without or people without cancer. But if you want to actually look at a cancer-derived cell-free DNA, of course, it has to be a DNA which is having a particular mutation, which is generally associated with the tumor and which are not usually observed in normal people. So liquid biopsy, although it's a recent technique, but people had already predicted that there could be this cancer-derived DNA which are present in blood. It was almost in 1930s where it was predicted, but at that point of time, people did not know how to take those DNA out from blood. But now there are techniques available, and which is quite commonly employed in particular rare kinds of cancer which are encountered in the clinics. So there are two mechanisms which have been proposed for why cell-free DNA should exist in blood. So one is the active hypothesis, or the other one is the passive hypothesis. Active hypothesis says that cancer cells release oncogenic DNA in blood to affect the transformation of normal cells. And the passive hypothesis is this: the cancer cells, as they are growing, there are also a subset of cancer cells are also dying. So they are undergoing necrosis process or apoptotic process. And when this is happening, these nuclear DNA or mitochondrial DNA, which are released in the bloodstream, remain in the circulation for a short period of time before they are degraded. So the concept of liquid biopsy that we practice in today's context is based on this passive mechanism, which tells that as the tumor is growing, they are shedding these DNA which containing mutations, patient-derived or patient type or patient-specific mutations, and these are generally associated with the somatic changes that are happening in the particular tissue. And once we have a technique to extract this cell-free DNA, we can detect these cancer-specific changes in blood. So what are the different kinds of mutations one can check? It is something called as point mutation, which you can see in the blood, in a DNA which is obtained from blood. Something structural rearrangements, particularly for, say, BCR ABL transfusion in CML, one can check that. You can also check methylation in particular tumor suppressor genes. And of course, finally, one can also check the presence of viral DNA. So these are some of the things that one can easily make out. So if you look at the applications of liquid biopsy in cancer management, there are three major applications. One is the diagnosis of a cancer. It helps in diagnostic application. The second one is the prognostic, whether there is any disease relapse or recurrence of the disease, or whether the patient is actually responding to a particular treatment. And the third one is predictive, whether a person qualifies for a particular treatment. And this is more applicable for targeted therapy in cancer. And today's talk, I will try to give an example on targeted therapy in lung cancer and the kind of work that we have done using this liquid biopsy in lung cancer cases. So the example that I'm taking up today is how liquid biopsy can be employed to understand targeted therapy in lung cancer. And of course, as some of you already know, targeted therapy is basically the TKI therapy or tyrosine kinase inhibitor therapy that we talk about, and which is most applicable for the non-small cell lung cancer, which is the largest category in lung cancer cases. Almost 85 to 90% of the lung cancer cases belong to this non-small cell category. So if we want to talk about targeted therapy, I would like to quote this man. He has nothing to do with cancer genetics or cancer research, but his quote can be very well applied in cancer or lung cancer therapy. So what he said was, we tend to overestimate the changes that occur immediately, but we tend to underestimate the changes that will occur in the long term. So in case of, he has given example of 2 years and 10 years, but in case of lung cancer, I think it is about 15 years what we have seen the change. So the change is moving from conventional to targeted therapy. And nowadays, in most of the cases where people are treating cancer patients, I would like to request the participants to kindly turn off their mics because there's a bit of disturbance that is happening. Thank you. So in case of lung cancer, there is this conscious shift from conventional to targeted therapy. And TKI therapy is something which is very commonly employed in all the medical oncologists. The first thing that they want to look at is the TKI therapy, and then nowadays immunotherapy is also quite common. Of course, these are expensive, but they have their own advantages, particularly on the side effects that you see in chemotherapy or radiation therapy. So how did it happen? These are two landmark papers that actually led to this identification. This is a paper that came in New England Journal in 2002, where they said that regardless of whether a person is being administered a single therapy or a doublet therapy, there is no change in median survival, there is no change in time to progression, and it does not offer any significant advantage over other treatment in non-small cell lung cancer. So then people were looking at what is the reason, and then in 2004, another paper came that TKI therapy, or the activating mutations in EGFR were actually making people responsive to a particular drug called geftinib. The geftinib was the first TKI or tyrosine kinase inhibitor which got approved, and that was approved in 2003. But quickly people felt, or people realized, or the clinicians realized that not all patients were responding to geftinib. Some were responding, some were not responding. So they found that there must be a genetic link to it, and they tried to understand what was that, and that was what is known as marking of patients. So what they found was patients who were responding to geftinib, which is the first generation TKI, or the tyrosine kinase inhibitor that came into the market, almost all of them had certain somatic mutations in the tyrosine kinase domain of this gene called EGFR, which is one of the important biomarkers in lung cancer. And they found that they were all carrying this mutation. And then the concept of tyrosine kinase inhibitor came up, and as we all know, it is a paradigm, or it's a great change that happened in lung cancer management. We have different kinds of tyrosine kinase inhibitors now in the market, depending on which gene they are targeting. If it is EGFR, we have geftinib, erlotinib, osimertinib. Osimertinib, it is a third generation EGFR. Then we have something alectinib, ceritinib, crizotinib, which are called ALK fusion. And if ROS is mutated in the patient, then crizotinib is again used for ROS. And then if there is a RAF mutation that is there, these dabrafenib or tremetinib are used. So all these drugs work on inhibiting the tyrosine kinase property of these proteins. And among all of them, almost 34 to 35% of the patients usually have EGFR mutation. So the most important target was found to be EGFR. So people looked at this and it was working very fine, but then it quickly became clear that people are also developing resistance to TKI. So when they started looking at the reasons for the resistance, again, they found that it was the EGFR gene again, which was the main contributor to the resistance of the patients. And just like those mutations that they found, which made patient responsive to TKI, the same TKI domain of EGFR was contributing to the resistance. Of course, it was another exon, and one particular mutation was actually responsible for this resistance. And people found that people were developing this, or this mutation was happening after the treatment. So initially, this particular mutation was termed as an acquired mutation. That means the mutation getting acquired after the treatment started in the patient. But lately, it has become clear that some patients have actually baseline mutation, means they already have this mutation even before they are subjected to TKI therapy, which is slightly unfortunate, because if they already have this, then they become ineligible for the treatment straight away. So this is the concept that led to the what is known as molecular testing in non-small cell lung cancer. Now, any person working on non-small cell lung cancer, any oncologist, if they diagnose a lung cancer case, and if it happens to be a non-small cell lung cancer, they recommend checking for mutations in these 4 exons of EGFR gene. Exon 18, exon 19, and exon 21, there are signature mutations, which are shown here with the red outline. These mutations are basically associated with sensitivity. So that means if a patient has any of these mutations, it's a good thing, because it is understood or it is confirmed that the patient will respond to the TKI. So that means the drug will be able to act on the EGFR protein. On the other hand, if they have a mutation in exon 20, which is that 790th amino acid, if it is changes from tyrosine to methionine, then there is a problem. That means the patient is developing a resistance to the drug which is being given. So that would mean that the patient is no longer reacting to the drug. Then of course they have to look for the second generation, the third generation inhibitors, which work even in patients who carry this particular resistance mutation. So the purpose of molecular testing in non-small cell lung cancer is to decide on TKI, of course, whether they are first eligible for this targeted therapy or not. Second, to monitor the emergence of resistance. As I told, this T790 mutation is usually an acquired one. Majority of the patients will have it after they are administered with TKIs. So to monitor how quickly or how late the disease is coming in them, or the mutation is developing. The conventional approach for that is the tissue biopsy, as most of the people do. But collecting lung tissue is very difficult. Part of it has to go to histopathology, and the remaining part can go for immunocytochemistry, and whatever is left is used for somatic mutations, checking for somatic mutations. Recently, this unconventional approach is being followed, which is liquid biopsy, looking at the cell-free DNA obtained from the tissues. And this is a preferred approach in recent years, and more and more labs are actually recommending the checking of these signature mutations. These 4, at least these 4 mutations are checked for patients. So there are different ways by which one can do liquid biopsy or checking for mutations. Of course, one straight approach is the real-time PCR. Then there is something called digital PCR, or it can be a chip-based or a droplet-based. What we do here in our university is actually the digital PCR. And then there is something based on beads, emulsion, amplification, and magnetics, beaming it is called. And then the final one is next generation sequencing the entire pool of cell-free DNA and looking at what all genes are mutated. All of these are very expensive, not affordable by the common man. Of course, NGS is really a lot more expensive compared to the other methods. Digital PCR is being followed for some cases, but in a research setup, I think the cost is much less. But in a private or in a commercial setup, I think it is very expensive. So the idea is to look for the feasibility of this test, whether it actually is able to detect or predict. And towards that, we have started work on liquid biopsy of lung cancer. And the next part of my talk, another 10 minutes I will try to explain how we do it in lung cancer. So digital PCR technique, it is the third generation PCR that we talk about or that we know. those who are well versed with the PCR technique, some of the people in the audience, I'm sure you have heard about the first generation PCR, which is a qualitative. Then we have this real-time PCR, which is a quantitative method, but it is done based on relative quantification. So you need to have a housekeeping gene, you need to have a standard and all of that. Digital PCR came in existence in 2011, and at the moment it is the latest and the most advanced PCR technology available in the market. And it relies on the same mechanism that is employed in real-time PCR. Only difference is that it allows you to do absolute quantification. Endpoint quantification, yes, but absolute quantification, and it does not require standard curve, or it does not allow relative, or it does not depend on relative quantification. So here the major difference between a real-time or a conventional PCR to digital PCR is the concept of partitioning. So one measurement is divided into 1,000 several discrete measurements. In a traditional setup, if you are thinking of, say, a reaction mixture of 20 microliter in one PCR mix, in digital PCR, what happens, this 20 microliter is divided or distributed into 20,000 individual droplets, water oil droplets, and each droplet may carry some DNA, may not carry some DNA. If they do carry some DNA, each droplet or each strand of DNA undergoes individual PCR amplification. So in a normal PCR, you have 20 microliter that undergoes PCR amplification million times as a single unit. In droplet PCR, 20,000 different PCRs happening at the same time. So the idea of this is basically to try and check or increase the sensitivity of the technique several thousand folds, and also to identify rare detection, rare events which are not possible to detect by, say, the real-time PCR or even by sequencing, Sanger sequencing. So digital PCR is similar to, say, next generation sequencing in terms of its sensitivity. So these are some of the techniques. This is the steps by which it happens. So we have droplet generation, then plate sealing happens, then, as I told, individual droplet undergoes a standard PCR in a normal PCR machine. And then these droplets are read by a droplet reader, and then finally the data analysis. So it requires about 4 hours to do this. And what we have in our institution is a bioread system called QX200, which works on the principle of droplet PCR, droplet digital PCR. There are other companies which make digital PCRs, but in India, I think Biored has the largest market. There are a few institutions which use a machine from Thermoscientific, which works more or less on the same principle, but they have this chip. So basically, they also distribute, they also partition, but there are individual grids where the DNA is amplified. Here, these are individual water oil droplets. So that's the major difference. And the working principle is same as we do in competitive PCR or in real-time PCR. So one set of primers and 2 sets of probes. Each probe is attached to a fluorophore. And the fluorophore is specific to the particular probe. If the probe is able to bind to the wild-type sequence, it will have different excitation. If the fluorophore is able to, or the probe is able to bind to the mutant sequence, it will have a different excitation. So generally, FAM is used for the mutant allele, and hex is used for the wild-type allele. So what is shown on the right-hand side is the 4 different scenarios that you can have. So these are given in different color codes. The black ones indicate the droplets. Each dot is a droplet, 20,000 droplets that I talked about. The black ones are the droplets which did not have any DNA in them after amplification. Because when the distribution happens, it is by chance. It usually takes care of the Poisson distribution in statistical parlance. So there is a possibility that some droplets will not have any DNA. The second possibility is droplets containing only the mutant template, which is shown here in blue color. The third scenario will be the droplets that are containing only wild type DNA or the good DNA. So that is shown in green color. And the one which is shown in orange are the droplets which contain both wild type as well as the mutant copy. And that is what is usually expected, because EGFR is an oncoprotein, and we all know that the changes happen in an oncogene is usually change in a single allele, which results in gain of function. I think one of the speakers, earlier speakers, did mention about how oncogene is generated from a proto-oncogene. In case of oncoproteins, usually the change happens in a single allele, and these orange droplets that you see are the droplets that contain both the wild type allele and the blue allele. So the more number of orange droplets, you will expect more number of blue and as well as green. So it is not a summation of these two, it is just the representative of that which are containing both the copies. So this is a snapshot of how it looks on the data. You can have a 1D plot, means you are looking at a single excitation of a single probe. Here it is shown as a blue color, so you're looking at whether the sample contains only positive the mutant probe. And then on the other side, it is the 2D plot where the same thing which is shown in the previous slide is shown here. You make 4 quadrangles and you have the black plots, you have the blue dots, you have the orange dots, and you have the green dots. So based on this, you quantify them, you can express in copies per microliter of blood. So that is the level of precision. You are expressing mutant copies per microliter of blood. So that's the level of precision that you see. So here are some of the examples that will show you. Unfortunately, the slideshow is not happening, so I had a little bit of animation done. But anyway, I'll try to explain. So we have done so far about 118 cases, about 84 males and 34 females. We have extracted cell-free DNA from them, and then we looked at 4 different types of mutations. G719S is in the exon 18. E746 to 750 is a 15 base pair deletion in exon 19. So 87 screens were done for G719, 88 screens were done for exon 19 deletion. T790M, it is a 69 screens were done. And the last one is L858R, which is a mutation present in the exon 21 of EGFR gene. About 80 screens were done for that. So in total, about 24 screens were done in 118 cases. And we did 4 hotspot mutation analysis. 3 of them are the ones that are associated with sensitivity to TKI, and 1 which is associated with resistance. And this is what it looks like. In our population, at least, we see majority of the patients carry a mutation in exon 21, which is L858R. Out of 88 screens, about 57 are positive. Whereas in case of exon 18 and exon 19, the number is almost identical. Exon 20, surprisingly, we did not expect this, the patients to have baseline mutation, but almost 8 patients had already this baseline mutation in them, which means that they are already carrying this mutation. It could be a silent mutation, but since it is associated with resistance, generally, if this is shown in the patient, clinicians do not recommend TKI for that. And if you look at the non-small cell and the small cell category, again, it was surprising for us that small cell cancer patients who are not supposed to usually have this mutation, about 7 or 8 patients actually carried sensitive mutations. Fortunately, they were carrying sensitive mutations, means they were also eligible for TKI therapy. So about 5 patients had exon 21, which is shown in orange here, and about 3 patients had exon 18 mutation in small cell cancer cases. And this is a snapshot of total 118 cases. Unfortunately, the slideshow is not happening, so you can see those bars. These are represented as patients carrying 3 different mutations, patients carrying 2 two different mutations, particularly the one, if my cursor is moving, you can see this, this particular case where the patient has one sensitive and one resistant mutation. Same in this case. So that means they were carrying dual mutation, one sensitive and one resistant. The other patients, the shown here on these three patients, these are representative images, of course, they carried all the three sensitive mutations. And of course, some patients did not have any mutation, like these two shown here, they did not have any mutation. That means we have checked only EGFR, they could have mutations in ALK or ROS or RAF. This is how it looks like. This is an example of a predictive response. The one which is shown here in a circle, this is the patient DNA. What we are showing now is patient DNA and a negative control. So the negative control should not have any droplets, which is shown here. There is no droplet in this. I'll try to play this slide as a slideshow, and I think the animation will work, hopefully. So here, this is a patient cell-free DNA and negative control. The blue dots means they are carrying a mutation, which is here, it's written as T790. The bottom part of the image is the mutation or the wild type allele. All of them should have this, so that means it is correct. Negative control should not have any droplet, which is shown here. There is no droplet. So the droplets are with mutant DNA, and the droplets, the lower one is the droplets without a mutant DNA. Let me play another example. Here is again another example. Again, here we are taking a patient sample and a positive control. In the previous slide, I showed you the patient sample and a negative control. Here is a patient sample and a positive control. The positive control of the patient, both should have the green droplets, which we are seeing very clearly. If you look at the patient DNA, which is on the left-hand side, and the positive control on the right-hand side, and there is a yellow line here, which is not very clear, but anyway. So what you see here, a very large number of droplets. That means they are highly positive for this, and they are carrying a copy of about 47.1 copies per microliter of blood. It's a very high number. Fortunately, this is a sensitive mutation, so it is a good thing. So the result is patient is positive for this sensitive mutation, and the conclusion is the patient is eligible for TKI treatment. Let me take another example here. So this is a sample which is negative for the T790 mutation. Here again, there is only green droplets, there are no blue droplets. So that means the wild type DNA is present, the mutant DNA is absent. So the conclusion is the patient is negative for this T790. So that means the conclusion is the patient is eligible for TKI treatment. And this is another one, which is again prognostic application, which is very important. So this case, we observed the patient for about 2 years, and every 6 months we collected the sample, and we wanted to see at what stage they are developing the resistance. So the first one, second, third, fourth, fifth. So like this, these are follow-ups. So if you see here in the first 3 columns, there is no. The first one is of course negative control, and then 5 follow-ups here. There are no droplets, but if you see here, towards the end, the number of droplets, the blue droplets are increasing. So initially in the 4th follow-up, they had a slight number, and then the 5th follow-up, they have more number. So that means the patient is developing the resistance. So these are the follow-up samples that we have, and the 4th follow-up onwards, we started getting this mutant DNA present in their blood. So we are looking at the presence of this mutant in their cell-free DNA. So that means at the 4th follow-up itself, they are showing the presence. At this point, the patient may not have clinical manifestations already, but the doctor can already predict that, yes, this is something the patient is likely to develop resistance now, so the treatment has to be stopped. And same thing happened in the fifth follow-up, increased in number. Of course, the green droplets remain same, so that means the wild-type DNA should be present in all of them, so they have this. So the prognosis is from 0 to 0.14 to 1.4 to 3.4. There was a progressive increase in the number of mutant droplets, and this allowed us to detect the emergence of resistance, and liquid biopsy could help us do this very effectively. So coming to the conclusion, the liquid biopsy is very effective technique. It can be repeatable, and it's a minimally invasive or a non-invasive technique that allows real-time monitoring. It has greatly enhanced impact of target therapy, and in non-small cell lung cancer, liquid biopsy is routinely followed. People are using ddPCR or NGS-based approach. And as I told, clinicians can monitor the emergence very easily, even before the cancer has grown to a certain size, because the imaging techniques that allows the detection of cancer requires the cancer to grow or the tumor to grow to a certain size. Liquid biopsy can do it even before the tumor has grown. And because it is so sensitive, it can be done with unparalleled precision. And many commercial companies in the country are offering ddPCR services. Of course, the cost is really expensive. For one set of EGFR panel, they charge about 22,000 rupees, which is very expensive, but maybe alternative techniques. We are also doing certain alternative techniques using the same cell-free DNA, and we are trying to see whether we can avoid using a ddPCR machine, whether we can use some other ways of doing it. We are developing those things. So this is the cancer genetics team. These are my clinician partners from the pulmonary medicine department, Dr. Giridhar and Dr. Rajesh. Incidentally, both of them have, Dr. Giridhar has already completed his PhD with me here, and his topic was also something related to that. Dr. Rajesh is completing this year, and this is the students, JRFs and other students who are involved in this. And finally, this is my ddPCR setup. There are 4 components to this, which is I showed you in one of the slides. This is the sealer, and the generator is here. It is not visible, and then the PCR machine and the reader. So thank you very much. I hope I could stick to the time that was given to me. I once again thank the organizers for the opportunity, and it was a great opportunity for me to interact, and hopefully we'll be able to interact in person at some point of time. Thank you very much.
Thank you, sir, for the elaborative talk on liquid biopsy in cancer management. Now I request participants. Yes, yes, sir.
Yeah. Is there a time to take any questions? If there is anything, I'll be able to take. I guess I have 2 more minutes to take any questions.
Sure, sir. So now I request participants to post queries on the chat box.
Thank you, Dr. Anirban, for your wonderful. Thank you. The quality inputs that you have given. So this is an eye-opener for any of the researchers. Thank you.
Thank you.
Questions?
Thank you very much.
Sir, there is a question for you.
Yeah.
Any studies on breast cancer, sir?
Yeah.
From Harendra Kumar.
Okay. Liquid biopsy is actually quite useful in breast cancer as well. We are doing, particularly on triple negative, because other breast cancers are relatively easy to manage. So triple negative, we are looking at certain signature mutations, and we are looking at peripheral blood, particularly for the response to the treatment that is done. So we have initiated a few studies on breast cancer. We are also doing on oral cancer. So in oral cancer, what we are doing is we are trying to use saliva as a source of cell-free DNA instead of blood. And if we did a bit of preliminary study on that, and we are able to find tumor-derived DNA in saliva. So if saliva is available, then we can go into a completely non-invasive method instead of minimally invasive. So these two projects are happening in the group.
Thank you, sir. Sir, there's one more question from Dr. Usha. Can liquid biopsy replace conventional biopsy in future?
Yes, the answer is yes. I think very soon people are, more and more, if you look at the cancer treatment strategies, people are moving away from conventional therapy. And I think 10 years from now, probably chemo radiation will become obsolete. One of the hindrances in therapy is the exorbitant cost in immunotherapy or radiation or targeted therapy. People are developing cheaper drugs, so it may become affordable. And if they are affordable, people will definitely go for targeted therapy or these particularly immunotherapy. Now, if similarly, in case of diagnosis or prognosis, I'm sure liquid biopsy will. In fact, liquid biopsy is a standard technique which has been approved by FDA now for CML, for looking at the BCR-ABL fusion. There are a lot of kits available from Biorad, which are routinely used. So more and more people are getting into it, and I don't see any reason why they can replace. In fact, there are studies where people are doing liquid biopsy to subtype lung cancer cases. So if that is possible, then people may have to do away with conventional histopathology. But I don't know if there are histopathologists in the team, they may not agree to my idea. But at this moment, histopathology is the gold standard. People need to look at the tumor tissue to actually tell that there is a malignant transformation. But there are plenty of cases where we know that physiological changes or morphological changes in the cell not necessarily associated with genetic changes. And at the end of the day, cancer is a genetic disease. Whether we accept it or not, everything that happens in cancer are genetic changes at cellular level. So these techniques which can detect genetic changes in blood can truly replace. Not in the near future, but definitely in the long term.
Thank you, sir. Sir, there's one more question. What's the positivity detection rate difference from lung biopsy per se and cell-free DNA?
It's a good question. We did a study on comparing our ddPCR detection with the traditional. So what happens in lung cancer? Generally, people look at immunocytochemistry. And in immunocytochemistry, they particularly look for, say, overexpression of EGFR protein. And if there is overexpression of EGFR protein, then they look for these activating mutations. So whatever mutations that I showed, these are called activating mutations. That means they are involved in converting an inactive EGFR into an active EGFR oncoprotein. Now we looked at the ability of ddPCR to detect these changes, and we compared with sequencing, standard Sanger sequencing. We found that, surprisingly, samples which Sanger sequence could not detect were positive in ddPCR. So that means the sensitivity is several folds higher, and one must also understand that these mutations are rare mutations. Generally, they are present in 0.101% to 0.1%. ddPCR enhances the ability to detect this by almost 330 folds. That means from 0.01%, it can enhance to almost 33%. If you want to compare, of course, PCR is a lot more sensitive. Some people might argue that whether it is overrepresentation of the. So that call is to be taken by the medical oncologist, because we are talking about, we are not looking at normal people. We are looking at patients who have been clinically diagnosed with cancer. So for them, the whole idea is to look at whether they are eligible for a better reduced cycle. So the sensitivity is a lot more higher, and I believe comparison should happen between the NGS methods which are used for detection. Traditional histocytochemistry does the protein expression level, and only if protein change is better, then only the protein expression is more, then only the genetic change.
Thank you, sir. So another question, prognostic and diagnostic role of microRNAs in breast cancer.
Yeah. If you are talking me whether liquid biopsy can help in that, the answer is yes. I'm not getting into how microRNAs are responsible in breast cancer initiation or progression. But if the question is whether liquid biopsy can help in diagnosis or in prognosis of breast cancer using microRNAs, the answer is yes, we can do
that. Thank you, sir. Thank you all the participants. Now we'll break for lunch. We'll resume back at 2:30 p.m.
Thank you. Thank you very much.
So we'll start. Good afternoon, one and all. I welcome back all the participants for the CME. Current trends in cancer biology for the post-lunch session. So now I request Dr. Azim Mohiuddin sir to present on treatment modalities in head and neck cancer. I would like to say a few lines on sir. Dr. S.M. Azim Mohiuddin sir, Professor, Department of Otorhinolaryngology and Head and Neck Surgery, and Director of Research and Innovation, Sri Devaraja's Academy of Higher Education and Research, Tamaka, Kolar. Sir is honored with University Gold Medal from Command Hospital Air Force, Bangalore in the year September 1995 for MS Otorhinolaryngology. Sir is also honored with Fellow of International College of Surgeons in the year 2017, awarded Fellow of American College of Surgeons in the year 2018. Sir is elected as member of National Academy of Medical Sciences in the year 2019. To his credit, Sir has got 84 publications in national and international journals with over 474 citations. Sir is in the process of writing 2 chapters, Thyroid and Salivary Gland in an ENT textbook. Sir has got Gurumurthy Gold Medals 3 times for best paper among teaching staff in State and South Zone Otorhinolaryngology Conference. That is in the year 2004, 2006 and 2007. And also for the Sake of Honor Award by Rotary Club during June 2006. Sir is also honored with Bharat Seva Ratan Gold Medal Award during July 2013 by GPRA Foundation, Chennai. Sir has got teaching experience and worked as teaching faculty at various head and neck, otorhinolaryngology and oncosurgery CMEs all over South India. Sir is convener of Head and Neck Oncosurgery Fellowship Program and Oral Oncosurgery Fellowship Program at Sri Devaraja's University, India. Sir has given guest lectures and demonstration surgeries in various institutions all over South India. Sir is a selector and examiner for fellowship course in Head and Neck Oncology at Kidwai Memorial Institute of Oncology, HCG Bangalore and Christian Medical College, Vellore. So sir's areas of interest are voice conservation surgery in advanced laryngeal cancer, pediatric airway emergencies, epigenetics in oral cancers, locally advanced thyroid malignancies, locally advanced salivary gland malignancies, compartment resection in oral malignancies with infratemporal fossa involvement. With this brief introduction, I welcome you, sir. Over to you, sir.
A very good afternoon to one and all. At the outset, I would like to welcome you all for this talk, and I would like to thank Professor Shashidhar and his team, the organizers, for giving me an opportunity to talk on my favorite topic, that is cancer. The topic given to me is treatment modalities in cancer. I have slightly modified it to head and neck because I'm a head and neck surgeon. I will trespass a little bit into other territories briefly, but my main talk will be on head and neck. And one thing I would like to confess, being a surgeon, I may stray into more of surgery, but I'll try to limit that and come back to diagnostics and chemotherapy. So as we all know, cancer is a change in tissue where programmed cell death is lost. The cell division becomes mitosis instead of meiosis, and there will be uncontrolled growth of the affected tissue at the cost of the host. We all know this. These are the areas I deal with in cancer. The entire head and neck, oral cavity, pharynx, nose and paranasal sinuses, larynx, hypopharynx, and thyroid and parathyroid glands, salivary glands, adnexal cancers, and metastatic cancers in head and neck, and of course, reconstructions. When we say head and neck, this is an important region because it is a compact region where a lot of important blood vessels and nerves are clustered in a small region. They are functionally important areas. Any compromise here can affect the quality of life and the basic functions of survival. And it is also an aesthetic region. So the problems with head and neck cancers is they affect breathing, chewing, swallowing, speaking, eyesight, endocrine functions, lubrication of food, hydration, and cosmetic appearance. Down the edges, cancer used to be thought to be a curse, an incurable mutilating disease. In fact, patients used to be shunned, and there was no hope till the mid-19th century. You can see the sort of cases who come to us. Even today, 80 to 85% of our patients present with advanced disease. All the more important for us to catch them early so that the outcome is better and the treatment is less aggressive. You can see inoperable disease here. When we talk of cancer management, it started in the last 2 decades of 19th century. The evolution progress involved surgery, radiation, chemotherapy, and biological, that is molecular intervention, and of course, prevention, surveillance, and palliation for advanced disease, and special gadgets. Each of these I'll be discussing briefly. So when we come to surgery, it started with resection. Anything that you don't like, get rid of it. So in the process, we used to have mutilating surgeries with very high mortality and morbidity, and sometimes discouraging results. You can see a picture of Sushruta. Way back in those days, he started surgery. And cancer and reconstructive surgery of the nose was one of his achievements. Just to go into a few surgeries, earlier, maybe in the last century, thyroidectomy was so morbid, it was banned by the church in Europe. However, people like Bill Roth, Cocker, and Halstead made this surgery safe and popularized it by the end of 19th century. In fact, in 1909, Sir Theodore Cocker was awarded Nobel Prize in medicine for his excellent work on thyroids. You can see this gentleman. A surgical photograph of thyroidectomy we have performed. You can see the huge thyroid gland, and you can see the recurrent laryngeal nerve and parathyroids preserved. The carotids, this tumor was in fact in close relation to the carotid vessels. You can see the sort of cases we get. Anywhere else, I don't know how many people would have touched it in other countries. But again, and this lady had pulmonary metastasis when she came to us, but it was differentiated cancer. So we operated, gave her adjuvant treatment, and she survived for 3 years after this. This was a metastatic node, which weighed 750 grams, single node. Coming to oral and oropharyngeal cancers, they involve ablative resections. Means, since it's oral cavity, you can see them from half a kilometer and say what they have undergone many times. The neck dissection used to be radical when it started in 1906. And of course, the reconstruction when it started was primitive and inadequate. It has improved over a period of time. And when they started it, because of the high mortality and morbidity, it was nicknamed commando. If you give a breakup, it's combined mandibulectomy, neck dissection, and oral or oropharyngeal resection. You can see the advanced cancer. All this is operable today, and you are seeing the surgical photographs as well. The resected specimen. This is something we excel in. A tumor like this was considered inoperable till 2007. 2007, a gentleman Liao in Taiwan started with all this what we call compartment resections, and so did we, and it's published in European archives. This is it. Neck dissection, when it started in 1906, it was started by George Kreil. It used to be radical. That is, they used to resect all the lymphatics and lymph nodes in the ipsilateral side of neck en bloc. So in the process, all the fibrofatty tissue and fat used to be removed just to make sure all the lymph nodes, which number 300 on each side of the neck, come out. In the process, few non-lymphatic structures like accessory nerve, internal jugular vein, and sternomastoid used to be removed. This is the extent of resection in the neck. We have 5 levels laterally and 2 levels in the midline of the neck for lymph nodes. A neck dissection, radical neck dissection in progress. You can see the bare carotid artery there. Similarly, laryngectomies, when they started, it was only total laryngectomy, which involved permanent loss of speech, relatively high mortality. It was a morbid surgery. It was made safer by Bill Roth towards the end of last century. So these are Bill Roth's contributions to surgery. A total laryngectomy, the surgical photographs, you can see a huge hypopharyngeal tumor for which a total laryngectomy has been done. And this involves permanent loss of voice. Coming to radiotherapy, it started in 1898 with Madame Marie Curie and her husband Pierre Curie. When they started for medical use, it was mainly two-dimensional radiotherapy. It had its toxic effects, relatively poor tumor control, and over the time, safer isotopes and safer techniques evolved. We'll be visiting it. So the associated advances, in addition to surgery and radiotherapy, brought in safe surgery, safe surgical techniques and antibiotics, radiotherapy techniques which were safer and more efficient, safer anesthesia, and safer replacement of blood and its components, advances in reconstructive surgery, good nursing care, and advances in surgical gadgets which made life much easier for us. Similarly, in radiotherapy, the evolvement of digital radiographs, ultrasounds, computerized tomography, magnetic resonance imaging, angiographies, the positron emission tomography, functional MRI now, and isotope scans brought in a lot of advances and made treatment much safer and much more effective. These are the stalwarts in head and neck surgery, Hayes Martin and George Kreil. Now, in the 70s, people started thinking, do we really need to do a radical neck dissection and sacrifice the three important non-lymphatic structures mentioned here? So, Suarez and Boca, later followed by John Ballantyne and Bayer, started saving the accessory nerve and wherever possible, internal jugular vein and sternomastoid. We call this modified radical neck dissection. It is the same extent of resection as in radical. However, the non-lymphatic structures, if they are not directly involved, are saved, giving a better quality of life with the same tumor control. You can see the surgical photograph. And in the 80s, people like Jesus Medina started thinking the lymphatic drainage of neck is predictable. So in case it's N0, that is no palpable lymph nodes or just single lymph node, can we do less than a full neck dissection? So came the era of selective neck dissections. These are the two gentlemen who popularized it. In fact, Jatin Shah was the former president of American Head and Neck Society. You can see the surgical photograph and the post-operative photograph of a patient who underwent selective neck dissection. In oral cancer, we do supraomohyoid, and you are seeing the result, hardly any deformity, everything preserved. So these are the various selective neck dissections we do depending on the primary. In larynx and hypopharynx, we do lateral or posterolateral. In thyroid, we do central compartment. In oral cancer, which is most common in our country, we do supraomohyoid, that is levels 1, 2, and 3. And of course, endoscopies brought in, again, a lot of benefit. Rigid endoscopies, fiber optic endoscopies, contact endoscopies, where we use particular wavelength of light, narrow band imaging, etc. You can pick up the subtle changes in the tissues and vascularity, subepithelial vascularity, to know the extent of tumor. And endoscopy making life much easier. You can see a telescopy directly. Microlaryngeal surgery in early cancers. A laser can be used along with microlaryngeal surgery. The fiber optic endoscopies, be it laryngoscopy, bronchoscopy, esophagoscopy. These are the sort of tumors which can be removed endoscopically, preferably with a laser. And he is the gentleman who is called father of upper aerodigestive tract rigid endoscopy, Dr. Chevalier Jackson. So in endoscopies, we can do a biopsy, breast cytology, bronchial washings for malignant cells, stenting, dilatations after treatment, etc. Then came the era of mandible conserving procedures. It's not necessary that you always sacrifice the mandible in oral cancer. The tumor may just be abutting it, not invading. In fact, we did a study where we found hardly 26 to 30% of resected mandibles actually have invasion. The others, it's for surgical margin. So can we conserve the mandible? Where you have a dentulous mandible and tumor not going to the alveolar sockets, you can remove the alveolar part of the mandible and preserve the lower rim, maintaining bony continuity. Or you can cut open the mandible, swing it, remove the tumor in the floor of mouth or in the tongue, and fix back the mandible. So preserving this helps in viewing, in initiation of swallowing, as well as better cosmosis. A surgery where we have done marginal mandibulectomy, and the extent of resection shown by the red line. And similarly came voice conservation in laryngeal and hypopharyngeal cancers. We have a series of surgeries Since this is biochemistry conference, I will not go deep into it, but I would just like to briefly tell these involve partial laryngectomies, and partial can be horizontal partial in supraglottic or vertical partial in glottic cancers. They can involve subtotal laryngectomies like supracricoid laryngectomy. And very advanced tumors, if the interarytenoid region is free, we can do what we call near total laryngectomy, where only one functioning arytenoid we preserve, and that is enough for speech. However, these near total laryngectomy patients require a permanent tracheostomy. And I'm proud to say my teacher, Dr. Sultan Pradhan, brought this procedure to India. It was introduced by Bruce Pearson in 1980s. This is the extent of resection in supraglottic laryngectomy, which is horizontal partial. The extent of resection in vertical partial, which for glottic tumors. All T1 and T2 tumors can be resected safely this way. A vertical partial laryngectomy. Then came the era of near total laryngectomies. You can see the extent of resection. This is a schematic diagram where most of the larynx is removed, retaining one functioning arytenoid, and a shunt is created with the remnant of larynx opening into the pharynx, and that is guarded by the functioning arytenoid. They, however, require permanent tracheostomy. A near total laryngectomy, which we have done, that's a stent which we keep for 2 or 3 days after the surgery. This was a video actually to show how they talk with their own voice following a near total laryngectomy. In fact, it's called maintenance-free biological shunt. Our publication, 137 cases of near total laryngectomy, published in Laryngoscope. Similarly, people started doing conservation surgeries in thyroid cancer. Like if the tumor is low risk, lesser than 4 centimeters in greatest diameter and age of the patient less than 45 years, we can remove only that lobe provided there is no extrathyroidal spread, metastatic lymph nodes, or it's not aggressive variant stuck to the important tissues. And then came the help by magnification and optics. Thanks to that, we can do laser resections, transoral surgeries, transoral laryngeal laser resections. We can use the same laser for very early or very advanced tumor, either for resection or palliation, what we call photodynamic therapy. We can salvage small recurrences endoscopically. And just to go a bit further, with these endoscopic surgeries come in robotic surgeries, especially in difficult to access area like base of tongue. I would have to swing the mandible or do a pharyngotomy to reach the area. However, with laser, the anterior tumors are resectable. With robo, even the base of tongue tumors are resectable without really cutting the mandible. And also with robotics came in distant access surgery. You can do a thyroidectomy without a neck scar. You can come in through the axilla or through the mouth to do a thyroidectomy. It gives you magnification and precise tissue handling. That's a photograph of Da Vinci Robo. This is a laser surgery. You can see a small T2 tumor of the oral cavity. Sorry. T2 tumor of the oral cavity in this photograph, the same being resected by laser. Another tumor after laser resection, you can see. After laser resection for small tumors, we don't do reconstruction. It heals without much fibrosis. The distant access surgeries can be thyroidectomies, submandibular gland resections, and neck dissections. And you can approach this transaxillary, transoral, the hairline approach, or bilateral axillobreast approach. All this avoids the scar in the neck. A robotic surgery in progress for base tongue. These were all the advances in surgical gadgets. Electrocautery, which can minimize bleeding, harmonic scalpel, again minimizes bleeding and more precise tissue cutting with less collateral damage. The radiofrequency knife, ligashore to achieve hemostasis, the USA in neurosurgeries, laser, robo, surgiform to control massive bleeds, nerve monitoring in areas like recurrent laryngeal nerve, and gamma probe. With gamma probe came the era of sentinel lymph node biopsies. These are all the, this was a diploparotid tumor, which we had resected. A parotidectomy, you can see the entire facial nerve saved here. All the batches. After resection, you can see the nerve saved pointed by the hemostat. In this case, it was a diploparotid tumor. You are seeing the tumor just behind the teeth. We had swung the mandible to approach the tumor and got it out. A microlaryngeal laser surgery. This is the laser machine. And this is usually coupled to the ZIS operating microscope. A harmonic scalpel. And this is nerve monitoring during surgery. And to save the parathyroids, we can use indocyanine green, and the fluorescence caused by it can be used with infrared cameras, infrared monitors, and we can spot the parathyroid. That white spot encircled by red is parathyroid. A sentinel lymph node biopsy, many times lymph node metastasis does not occur. And in these cases, can we avoid neck dissection? The answer is sentinel lymph node biopsy. We inject technetium-99, and it's usually, we inject around the tumor, and the first lymph node or group of lymph nodes to pick it up is called sentinel lymph node, which can be tracked by gamma probe. You harvest the lymph node only if it contains metastasis on frozen section, go ahead with a neck dissection. Otherwise, avoid neck dissection. This is called super selective neck dissection. And then came the navigation techniques, particularly useful in paranasal sinus surgeries. Advances in radiotherapy involve safer isotopes, safer delivery of radiation, three-dimensional radiotherapy now. What we have, intensity modulated radiotherapy, image-guided radiotherapy, etc. These are more precise. They preserve important organs. They minimize the superficial damage, and we have radioprotective shields now. So came the advances in technique of radiotherapy, where you can extend the fractionation. That is, each fraction is each sitting of radiotherapy. You can extend it over a period of time, or you can reduce the duration by hypofractionation. We have something called accelerated hyperfractionation, where you give the usual dose is around 200 centigrade per day, 5 days a week. You can reduce it to 180 centigrade, but give it twice daily. We call it accelerated hyperfractionation. The brachytherapy, where you use radioactive implants, gamma knife, cyber knife in delicate areas. I've already mentioned IMRT and IGRT, which give maximum dose to the tumor and minimize the dose to surrounding area. And now we have proton beam. The IMRT in progress, you can see it's three-dimensional. You can even alter the shape of the beam according to the requirement. Proton beam treatment. Similarly, came advances in chemotherapy or medical oncology. There are knowledge in cell cycle, the cell checkpoints, markers for apoptosis, photosensitizers, combination therapies in medical oncology, like you can combine taxanes along with cisplatin, and you have various combinations for lymphomas. The targeted therapy, where we use monoclonal antibodies. For example, in head and neck cancer, we use monoclonal antibody targeted against extracellular ligand of epithelial growth factor receptor. We call it cetuximab. So these are targeted therapies. Nanotechnology, where you can deliver the medical, that is chemotherapy drug to the required area. You can use viral vectors for drug delivery. So all these are in progress. The various cell checkpoints, The common checkpoint being junction between G1 and S phase, and the other between G2 and M phase. The cisplatin acts on the checkpoint between G1 and S phase, thereby inhibiting, rather altering DNA synthesis. And taxanes, the second, the another first-line drug, it acts on the junction between G2 and M phase. Similarly, you have a metaphase checkpoint. Coming to cisplatin, it's the first-line drug in head and neck cancers. It acts on the junction of G1 and S phase, prevents synthesis and brings about DNA adducts. It also has a weak alkylating effect. Can be used as a single agent or in combination with either 5-fluorouracil or with taxanes, the paclitaxel or docetaxel. It has synergistic action with radiation. The taxanes are the other first-line drug, paclitaxel and docetaxel. They act on the microtubules and effective at the junction of G2 and M phase of cell cycle. Again, can be used as single agents or in combination therapy. And now they are emerging as the first-line drugs. Coming to non-surgical organ preservation, especially in larynx, the credit goes to various people, but this lady popularized it, Dr. Arlene Forestier. And this is effective from T1 to T3 cancers, especially beneficial to T3 cancers. Definite prerequisites are there. And this was, her study was a landmark study which brought in organ preservation in laryngeal cancer. That's the study. Various chemotherapy regimens have come in, like HIT trial, EORTC trial, etc. So chemotherapy can be used as neoadjuvant, that is before the definitive treatment. For an advanced tumor, you give chemotherapy alone 2 cycles over a period of once in 3 weeks and see the response. If they respond well, go in for organ preservation. Otherwise, if still operable, go for surgery. Chemotherapy can be used as adjuvant, where you have resected the tumor. However, the histopathology shows aggressive tumor. So you can combine it with radiation as adjuvant. You can have organ preservation where you give concurrent chemotherapy with radiotherapy, like Arlene Forestier's study. In very advanced tumors which are incurable, you have palliative therapy, palliative chemotherapy. And you can also use it with targeted therapy, that is monoclonal antibodies along with chemotherapy, the extreme trial, for example. The targeted therapy targets specific molecular sites, like in oral squamous cell carcinoma, we have the extra cellular ligand of EGFR. This was popularized by Bonner. He showed a clear benefit of using targeted therapy with radiotherapy compared to radiotherapy alone. This is his trial. Now, the tyrosine kinase inhibitors are slowly making their way, particularly for advanced thyroid cancers and to a smaller extent for salivary gland cancers. We have a series of drugs, imitinib, erlotinib, etc. are used in various head and neck malignancies and in few locally aggressive non-malignant variants like chordoma. The sorafenib, lenvatinib, and vandatinib are specific for thyroid malignancies, particularly medullary cancer. Similarly, there are other targets also for chemotherapy, like mammalian target of rapamycin inhibitors, and also phosphoinositol 3 kinase inhibitors. We have various drugs like everolimus. However, their use has not become standard of care because of the high toxicity and limited role. There are advances in molecular biology and genetics, a huge area, thanks to decoding of human genome and the advent of epigenetics. Various tumor markers are there. For example, in breast cancer, we have HER2 targets, we have estrogen and progesterone receptor targets, similar various targets in lymphomas, and we have various intracellular pathways to target. And intercellular crosstalk can also be a future target for evolving chemotherapy. And of course, oncogenes and syndromes, the mutations and their classifications, karyotyping, gene sequencing, be it Sanger sequencing or next-gen sequencing, everything contributes to our planning of treatment. And in due time, with bioinformatics, we'll be able to easily identify what are the repetitive mutations in a given population and target them for our treatment. So also the miRNAs can be used for diagnosis as well as for treatment. The human genome, the potential sites, are not going to details. Here's again one of my mentors, Dr. Tapas Kundu. He's a senior professor in Jawaharlal Nehru Center for Advanced Scientific Research, the previous director of Central Drug Research Institute of India. And these are our other collaborators from molecular genetics. On the left side, you have Dr. Arun Kumar from IISc, Dr. Soumitra Das from IISc, and our own molecular scientist, Dr. Sharath. Our publications in collaboration with this gentleman in high impact journals. Now coming to tumor suppressor genes, we all know P53 has been nicknamed policeman or guardian of human genome. Similarly, P16 has huge role now, particularly in cell aging, cell senescence, and it is also a marker, yes, a surrogate marker in HPV-induced cancers. In our own college, our own biochemistry department, Dr. Susan is working on survivin, and there are various other tumor suppressor genes which can be targeted. P53 acts on both cyclin kinase, CDK2 and CDK4. This was a landmark discovery. In fact, mutation in P53 makes malignancies aggressive, be it squamous cell malignancy or be it thyroid malignancy. Now, P16, I told you, it's making its way into cancer diagnostics as well as treatment. It belongs to the INK4A family. It acts at G1 and S phase checkpoint and brings about cell aging. It has two ways of action, acts on both the retinoblastoma pathway as well as CDK pathway. Other various molecular markers have come into use. For example, in thyroid, we have BRAF, RET PTC, that is RET papillary thyroid cancer, galactase-3, P53, the telomerase reverse transcriptase, and P10, etc. Lymphomas and squamous carcinomas based on the CD types can have targeted therapy, and we can do simple immunohistochemistry to know which are the CD cells dominant in them. Similarly, the other tumor markers in neuronal tumors, the neuron-specific enolase, the synaptophysin, and in rhabdomyosarcoma, the desmin, and P16, the surrogate marker in HPV-induced cancers. So biochemistry has a huge role both in diagnostics as well as in tailoring treatment. And I would request in every tumor board, one specialist from biochemistry background or molecular genetics background has to be there. The other tumor markers, just to name a few, alpha fetoproteins in liver cancers, carcinoembryonic antigen, both in medullary carcinoma, thyroid, as well as colon, etc. The ERPR receptors in breast cancer, HER2 receptors in breast, prostate-specific antigen, that is in prostate cancers, the lymphocyte markers, serum calcitonin in medullary carcinoma, thyroid, serum thyroglobulin in well-differentiated thyroid cancers, the BRCA Epstein-Barr virus antibodies, and telomerase reverse transcriptase, just to name a few. The role for tumor markers is huge in the near future. Other biochemical markers to know the response, we have various cytokines, particularly interleukins. We are working on interleukin 6. Similarly, salicylic acid, tumor necrosis factor alpha, VEGF, etc. Our publication, this was a ICMR STS project led by Biochemistry Department with our collaboration, which is published in JCRT. Various other advances in molecular biology include proteomics, metalloproteases, oncogenes and proto-oncogenes, as well as stem cell research. Stem cell research is also being used in organ regeneration. So the advances in reconstruction include various flaps, various axial flaps. Axial flaps have their own definitive blood vessel and can be distal flaps harvested and brought into the surgical field on their own vascularity. Microvascular free flaps are when you take the flap completely out, divide of its blood supply, and anastomose the blood vessel in the flap with the blood vessel of the recipient site. These are all few examples. I'll not go into this detail. A pectoralis major, which is axial flap being harvested, and after reconstruction, same pectoralis major. This is pectoralis major with a deltopectoral flap, again axial flaps. A forehead flap used for reconstruction. A osteomyocutaneous flap, where we have removed the middle third of mandible along with the soft tissues. We have used pectoralis major from inside and deltopectoral from outside, along with a rib. Locally advanced tongue cancer, a radial forearm free flap after reconstruction. You can see the neo-tongue using a radial forearm free flap. A fibula free flap. This young girl had her mandible resected and fibula was used for reconstruction, and you are seeing the result. Similarly, there have been advances in rehabilitation, where you have removed the larynx completely. You have tracheoesophageal puncture processes where the patient can talk. You have bite gate processes after removal of half of the mandible. You have 3D printing for maxillary processes for maxillectomies. Similarly, you have osseo-integrated implants following a fibula free flap, and eye processes where you have done resection of the eye. Your TP processes, 3D printing of maxilla for reconstruction. A processes following maxillectomy, osseo-integrated implants. Various advances in pathology and biochemistry also help us a lot. Better staining techniques, immunohistochemistry, the serial step sectioning of lymph nodes, tissue culture and cell lines, the nuclear and molecular pathology. My two mentors, Dr. Gopinath and Dr. Ravinder Dev, our surgical team. Now the path forward in the future. The stem cell therapy, particularly for organ regeneration or for making the organ functional after a paralysis during surgery or during treatment. Genetic engineering and gene therapy, nanotechnology for drug delivery, robotics and artificial intelligence. In fact, we are in the process of having a MOU with a company, Big Gen, to incorporate artificial intelligence in the periphery, in the field, to diagnose cancers as well as few other diseases. The epigenetic blockers, for example, we have found out with our collaboration with Nehru Center that hyperacetylation of histones predisposes to cancer. We can use histone acetylase inhibitors and block it. Similarly, cloning and regenerative technology and targeted therapy in a bigger way. So, to be honest, there is a proverb, sky is the limit. In oncology, sky may not be the limit. We need to work in collaboration. We need to catch them early. We need to be aggressive in our initial approach, a multidisciplinary approach, and we need to have more research, be it in diagnostics, be it in therapeutics, or be it in reconstruction and rehabilitation. Thank you.
Thank you, sir, for the exemplary talk. And thank you for enlightening us on treatment modalities in head and neck cancer.
Thank you.
So now the forum is open for discussion. I request participants to post queries on chat box.
Pardon? Yeah, one minute. I would like to invite questions. Yeah.
As usual, my question is, you mentioned about new adjuvant and adjuvant.
Yes.
So, what kind of clinical selection you do for these two?
Yes. See, whenever we treat early cancers, the principle is single modality treatment, either surgery or radiotherapy. When you have advanced cancer, it is multimodality. Multimodality can be surgery followed by adjuvant. Adjuvant is when we eat dosa, we have chutney or palya. It is adjuvant. So along with surgery, if you use radiotherapy or radiotherapy along with chemotherapy, that becomes adjuvant. You may have a tumor which is advanced when the patient presents to you. You would not have adequate access to resect it. Important structures very close to it, or borderline inoperable. Or your waiting list may be too long. You need to put drugs on the tumor so the tumor doesn't progress till he comes to your table. So there we get the palya first and then have dosa. So we give new adjuvant. That is the adjuvant given before definitive treatment, in the hope that it either puts drugs on the tumor or it will downstage the tumor to some extent, making it more surgeon friendly or more operable when we go in for surgery. Similarly, I told you in organ preservation, you have concurrent chemotherapy, concurrent chemoradiation, where we use radiotherapy 5 days a week, and in the weekend, especially once in 3 weeks, we give chemotherapy. We call it concurrent chemoradiation. The study popularized by Arlen Forestier. However, you may have a tumor, especially in hypopharynx, which is already big on presentation. So you would like to know, and organ preservation cannot be done once it's eroding the cartilage. There are only surgical organ preservation, like near total in selected cases, can be done. So you would like to want a T3 tumor, if it's big in size, to restrict the field of radiation. You may want the tumor to shrink a little bit. You can give neoadjuvant chemotherapy. If it responds well, we call it partial response. At least 30% of the tumor should shrink. Then you can go in for concurrent chemoradiation. If it doesn't have partial response, if it's still operable, you can go in for surgery. Similarly, as a surgeon, if the tumor is big, reaching important area, I would try neoadjuvant chemotherapy in the hope that it will at least sanitize my surgical field. And if you go into literature, in the long run, they claim chemotherapy used as neoadjuvant, to some extent, it cuts down the chances of distant metastasis.
So the classification comes based only on the stage of cancer.
Yeah.
It doesn't depend on the modal environment, whether it is fixed.
No, any TNE classification will have 3 components. The T is for tumor, that is size, area, depth, etc. The N is for nodes. How many nodes? Is there an extra nodal spread? So all that is seen. M is for distant metastasis, either liver, lungs, bone, brain.
The modal involvement doesn't decide your.
It involves the treatment. For example, that's what I was saying. If a patient has come to you with advanced tumor, T2 or more, the chances of micrometastasis, even if node is not palpable, even if node is not picked up on ultrasound or imaging, chances of micrometastasis exceed 20% or more. So the area of lymphatic drainage you need to treat, be it surgery, what we call neck dissection, or be it including it in the field of radiation. So based on the staging, we plan the dose and thank you.
Thank you.
Yes, yes. Yeah. As I was showing our collaborative research with Jawaharlal Nehru Center for Advanced Scientific Research, we have had, I'll just show you that. We have had various publications on that. One was by Dr. Stephanie, it was her PhD thesis. It's on, her work was on nucleoplasmin 1, which brings about, these are the studies, it brings about acetylation of 300 And once P300 is done, it undergoes hyperacetylation. That itself induces further hyperacetylation, thereby bringing in massive carcinogenesis. It's published in Biophysica et Biochemica Acta. And similarly, we have one publication in the European Federation of European Biochemical Sciences Journal with the same lab. And this was our work on the changes in oncogenes, basically YY1, etc. Again with Nehru Center. Similarly, with ASC, we have worked on TSC genes, the methylation which brings in carcinogenesis. Even with our own molecular lab, the one in Journal of Cellular Biology, it's with our molecular lab. There are markers. The only problem in head and neck squamous cell carcinoma is its heterogeneity. We still don't have a definitive foolproof marker. There are so many proteins, so many markers which may be overexpressed or underexpressed. Now the way forward is identifying the repetitive mutations in a given population. The bioinformatics can be used for it. The next-gen sequencing can be used. And once we have that to target that population in field studies, and those who have those potential targets should undergo close follow-up, catch them young and treat them, and bring in drugs for them. Thank you.
Thank you, sir.
Yes. See, wherever the immunity is suppressed, the risk for any malignancy is high. More than head and neck, I would put it systemic malignancy, lymphomas. That risk is there, but COVID is, that immune suppression is over a very small period. So I would not blame COVID for it. But yes, your point is right. If you remember, we had COVID-associated mucormycosis. We operated 57 cases here and another 7 salvage surgeries, mutilating surgeries. Whether our use of steroids, our use of industry-level oxygen predisposed to it, or our population being mostly agriculturalists who have a lot of exposure to mucor spores. Was that the cause? We don't know. Was diabetes the cause? We don't know. But definitely, all our patients most of them were diabetics, and many of them were on steroids, and they were all COVID associated. There have been studies where they have found that glucose regulating protein 78 is altered in COVID, and that may be predisposing. And they have also found COVID affects the islets of Langerhans, thereby affecting insulin control. So whether that disposes to mucor, we don't know. But why only second wave had this? These are all questions to ask. And another issue is our use of steroids. That brings the hyperglycemic lack of control, or was it the GRP78? All that it requires longitudinal studies ideally, and the mucor died with second wave. No, it's only sporadic. So these are all obstacles we do find in research. Thank you.
Thank you, sir. No, sir. No queries on chat box. We'll close the session. Thank you, sir.
Thank you.
Good evening, one and all. I take this opportunity to propose vote of thanks on behalf of Department of Biochemistry, Sri Devarajaras Medical College. First and foremost, I pay obeisance to our late founder, President Sri R.L. Jalappa, sir. I would like to thank our patrons of Sri Devarajaras Education Trust, Honorable President Sri G.H. Nagaraj, sir, Honorable Vice President Sri J. Rajendra, sir, Honorable Secretary Anumant Raju, sir, Honorable Chancellor Sri Rajeshan Jagdale, sir, Honorable Vice Chancellor Dr. Pradeep Kumar Ji, sir, Registrar Dr. K. Prasad, sir, Dean Faculty of Medicine and Principal Dr. P. N. Sriramulu, sir, Medical Superintendent Dr. Sheila, ma'am, and Vice Principal Dr. K.N. Shashidhar sir. I also extend my thanks to other officers of the Academy. I thank Dr. P.M. Sriramulu sir, Principal SDMC, for his unceasing support and encouragement. I thank Dr. K.N. Shashidhar sir, Professor and Head Department of Biochemistry, and Vice Principal SDMC, whose motivation has been pivotal in the department's accomplishments. I thank distinguished luminaries for accepting our invitation as speaker and delivering insightful presentations. I thank you, Dr. Anil Ines sir, Dr. Akila Prashant ma'am, Dr. K.N. Shashidhar sir, Dr. Chimaraj sir, Dr. Anirban Chakraborty sir, and Dr. S.M. Azim Mohiddin sir. I thank all the participants of the event for making it a grand success. Last but not least, I thank the non-teaching, photographer, and teaching team members who are part of this program. Thank you, one and all.